Good morning, everyone. So we have currently bottom in sight, but we're going to let the ROV guys and the videographers go ahead and do all of the balance checks for the cameras and make sure we have the appropriate lighting. So we're going to be about here for about 5 to 10 minutes. If you guys want to come back in then, we'll come back and see. As you can see, we've reached the bottom here. We have a sandy, muddy environment with a blind lobster swimming by, or walking by rather. So we're just going to go ahead and give these guys a few minutes to set all up. So we'll be back then. Are we good to go, pilots? Great, thank you. And good morning, everyone. Thank you for joining in on this lovely Saturday morning. Here we've touched bottom at the base of a continental shelf off the straits, within the Straits of Florida, off the coast of more of the Keys than Florida any of itself.
You fix that?
And as you can see, we've touched down on some what looks like a sandy bottom with some coarse and fine-grained material. And you can see some of those little volcano peaks there, and that's likely due to burrowing from various crustaceans and other fish that they bury then into the sediments, and then as they push the sediments up out of their burrows, they form these like little volcano-like features.
They're bioturbated is what we usually call that. Ooh, we got a sea star. Nope.
Oh,
Brittle star.
star.
You can see his central disc, and he's got long legs. It's pretty neat. One of them shorter than the other?
Maybe, or maybe that's just our perspective. Oh, it looks like, yeah, one of the tips did maybe get nipped off.
No worries, it'll grow back.
No problem there. Can we get the lasers on to see his size? Once. Seems to be some sort of stem structure of something that maybe once lived there. Likely a sponge. Maybe some leftover spicules of a sponge. Oh, this guy's about 10 centimeters long with his arms. That's one of the larger ophiuroids that we've seen on any of these dives.
Yeah, they're usually only a centimeter or two.
Yep.
Yeah, got lots of bioturbation up here.
Yep.
There's a small shrimp.
And if you're joining us for the first time, I'm Kimberly Galvez. I'm from the University of Miami Rosenstiel School of Marine and Atmospheric Scientists as the geoscience lead here. And to my right, we have...
I'm Stephanie Farrington. I'm from Harbor Branch Oceanographic Institute at Florida Atlantic University in Fort Pierce. I am your bioscience co-lead. We got a fish.
Oh, yeah. Well, we've been exploring the depths of the Blake Plateau during this cruise. Unfortunately, we did have to escape some bad weather over the last week, so we were not capable of diving. But we're back now, and we're now on the southeastern side of Florida, so we're in the Straits of Florida exploring this territory here. This is previously unexplored areas. This was newly acquired mapping data that we did just overnight last night, and this is why we chose to dive in this location. It seemed to be very interesting that no one within 120 nautical miles had explored this territory. So we're definitely interested to find out what we discovered down here.
This is a Lehmanema, I think, which is a type of codling. I think he's a little nervous about our giant lights.
Yep. Seems to be taking in one of those little potholes. Wonder if he made that himself. I don't think so.
Yeah, I don't think so either. They're not. I don't think they're known for digging, but
just in the mood to hide. And we have some squid accompanying us. As you can see, those are the creatures swimming by the front of the camera there.
Shadow.
And the shadows. It's also a great shot of the overall sediments of what we'll likely be consistently seeing through this dive. You can see a lot of fine-grained material, and this is all calcium carbonate. And in the background, you can see a little bit of how fine grained and some coarser grained materials of the skeletal fragments within the sediments in itself. And this is all calcium carbonate, which means that it's composed of the same material that your bones are composed of, just in different mineralogical forms. And they are in size fractions, what looks like to be from mud size fractions all the way to coarser grained materials. And these are precipitated out of the water column in supersaturated areas where either organisms form their own skeletal structures, such as like shells or crabs or other small little forams that live in the water column. And then once they die, their shell fragments stay behind and become part of then the sediments.
We have something at maybe a Gorgonian or something at 6.
Oh yeah, just right kind of
Maybe a
under the fish.
hydroid?
The little stick that's sticking out there.
Yeah. Oh, it's a worm.
Serpulid worm, yeah.
Yeah. With
Amphipods?
yeah.
Those are large amphipods.
They are big. They're pretty neat though.
Yeah.
Look like they're wearing crowns.
Yeah.
The king and queen of the
Of this.
Serpulid or Sibillid or whichever one it is. I can never remember.
I think this one's the Serpulid one.
Serpulid. I always get them confused. It never ends.
Those are little sea bugs. One has 3 antennae, and the other one I think has 4.
Yeah.
I don't know if that's a different characteristics of possibly male and female or anything.
Or one of them fell off.
Yeah, maybe one of them fell off or got chewed off.
Or it's hidden behind his head. We can't see it.
Yeah. Well, thanks.
They're cute.
And the worm seems to be out looking for some food that comes by through the current systems. A sea star?
I think it's a
It's a pteropod shell.
Yeah. With something schmutzy. Oh, is that an actual living pteropod?
Yeah, no, I think, or is that a sea slug?
Oh, maybe it's a sea slug.
I think it's a sea slug. One of those dancing, I don't remember.
Nudibranch? Yeah, or
Holothurian.
Holothurian. Yeah, could be. Yeah.
Thank you, our videographer.
He's tiny. Yeah, it might be a sea cucumber. Maybe a relative of Nypniastes. They're usually red, though. We've got
you
a
can
lot of
see
dust.
by how the ROV touched down and it kind of kicked up a lot of that material. You can tell it's very fine grain in this area, so it could be easily transported if the currents were strong enough. And it's different than what we've been experiencing throughout other locations where we have more of that coarse grain material and that's heavier. It still can get transported by current systems, but not as easily.
Got a shrimp.
And these are likely what are also causing those burrows and those little volcanoes, the bioturbation underneath.
So this might be a royal red shrimp. This might be Pleodicus, which would be the royal red shrimp. If it is, there is a fishery for this off the coast of Florida. You can see its swimmerets. So Pleodicus robustus is my guess.
Ooh, a squid just totally dived straight for it. I don't think that was on purpose, though. We're seeing quite a few of these bioturbated little mound features of those tunnels that then are dug underneath the surface here. And those actually can be preserved in the geologic rock record. We see them quite frequently. in areas that now have been exposed above ground, whereas they were possibly precipitated and formed in the marine environment. And we can actually make out quite often what these bioturbations are. And underneath, if you're lucky enough for the tube, the complexity of that tube connectivity network down underneath the subsurface is preserved, you can actually see that also above the rock record there. Ooh, and that is a piece of debris. It looks like a
box or something?
Yeah, metal, maybe something to keep fishing gear in or something.
Or electronics?
Yeah.
What do you think it was?
A juice box. Okay. Oh, you can see the straw. Oh, what is that pink
we've got a
thing?
pink guy. That's probably a fish.
And you can just
9,
see
right
by
right in front of that tuft of
sediment being
sediment.
wafted up into the... And the squids are not really helping here.
Oh, nice. This is called I want to say a robin, sea robin. Let me Google that.
Being encompassed by the fine sediments wafted out. It looks eerie now.
Yeah, it's a type of sea robin.
Triglyday is what
yeah, Triglyday.
Tina was calling it. And just as much as we could see it, now we can't. I think those squids are starting to murk up some of the water as they touch down. This almost looks like it was intentional. This is just like a beautiful scene of like sediments
coming out of the mist
over it. Yeah. It seems now to have some of the sediment on it. He's walking on the seafloor.
Yes, he's got modified fins so he can kind of walk around on the seafloor, if you will.
Almost crab-like. Wow.
They're very armored looking.
Yeah.
It's not that uncommon of a look for deep sea critters. We have armored shrimps and stuff too. They have that armored look.
And it has, is that another type of modified fin for sensitivity?
I'm not sure what's going on.
I don't know what he has in front.
Or more of like a barbel, you know, like a sensitivity barbel. Looks like a mustache. A combing mustache.
We're good whenever you guys are to move on.
Well, he's able to move it.
Yeah. He has some spines falling along. Ooh, a sea star.
Yep.
I think it's another one of those cookie sea stars. And a lobster in the back or another shrimp. Something of the sorts.
Maybe Heterotella in the background.
Oh, in the background, yep, the sponge.
Yeah, we're going to zoom in on this sea star, hopefully. Maybe we'll be able to see underneath him. It looks like his tube feeder pushing him up off the bottom.
Maybe. Could be on the hunt for something. I can't imagine food is of plenty here.
No. Yeah, you can see his tube feet. Normally we see him pretty close to the bottom. He seems to be up on something.
Yeah.
So you can see his tube feet sticking out. So that's definitely a goniaster. I don't know past that. If Chris Ma's around, I'm sure he'll chime in. But in my notes, it's going down as goniasterity.
I think you're right. He doesn't look full, so we'll call him still Cookie Star rather than a ravioli. And I think that sponge in the background.
There
Yes,
he is, yeah.
please.
A small
Almost dead
shrimp.
center, yeah. We have some skeletal material underneath it if we can then look at
Oh
that
yeah,
shell.
so that's an
Ooh,
armored shrimp.
you don't say.
Yeah, right. Glyphocrangon, I believe, if I'm remembering correctly.
Blends in very well with the sandy bottom.
Yep. Yeah, this is Heterotella. Yeah, Glyphocrangon was the armored shrimp.
Oh, we got a little squat lobster inhabiting it as well.
Oh, nice.
With some zoanthids or no hydroids taking advantage of the stem and the current.
Yep.
Oh,
And some
oh,
amphipods and the racepods and the...
and zoanthids at the base
Yeah.
of this.
Cool, they're pretty clear. You can
Oh.
see them. Like, oh, did he catch something?
I think he was trying to.
Catch something, buddy. All right, so this is Hylanema, which is a sponge. It looks like a cotton candy ball on a stick. And the stalks can grow a couple of feet tall, the stalks and stems he stands on. They're actually siliceous, so they're so fine they can be used as fiber optic cable. However, that is financially unattainable, so it's cheaper to synthesize it. Yeah, it's a sponge. It's a hexactinella sponge or a glass sponge. They're pretty common in the deep sea. They sit up on this stalk to keep themselves out of the sediment. Yeah. We have one of their stalks at our lab we use for show and tell. And it's about 2.5, 3 feet long. It's pretty big.
Yeah, I've seen that one. It's nice.
Like twisted.
And we have some little like stalks poking up out of the ground there, out of the sediment. Not sure what they are. Maybe some leftover spicules, but they seem to be in little clusters. Oh, look, and a crab right at the base there too. Is that a golden crab?
Yeah.
Oh, multiple crabs.
No, I don't know what that is. He's white. Maybe it's a type. It's related to a golden crab. Which would be, I don't think it is a golden crab though. He's a little different looking.
Oh, that was quick.
That'd be Geryonidae is the family.
Ooh. Oh, and this seems to be an interesting tunnel here also.
Yeah, possibly Acanthocharis. Acanthocharis burrow, maybe? We saw an Acanthocharis as soon as we hit the bottom, which is the blind lobster.
Yep. So we are slightly south of our original dive plan transect, and that was more because it would have taken an extra hour to try to reach our exact point for our first one, and we thought just a few tens of meters to the south wouldn't have the worst impact and change, and we wanted to make sure we got plenty of bottom water time. So in case you're looking at the dive transects according to the dive plan, that's slightly off center there. That is not a at least unintentional mistake, so we'll see it slightly different there.
So it looks like we have somebody living in here. We have an anemone of some kind, maybe a burrowing anemone at 3 o'clock.
Yeah.
And then somebody living in the little hole there. I'm not sure.
It looks
what
maybe
it
like
is.
one of those lobsters, maybe.
Yeah, it could be. It's kind of pink.
Yeah. Are the lasers on?
It can also be a snapping shrimp. They like to burrow out here too. If they'd be really tiny.
Yeah. This anemone is really translucent. It looks like does it have something in its tentacle? I'm not sure.
Maybe. Hi, who's calling in?
Hi, this is Christina.
Hi, Chris. How are you?
Pretty good, ready for another interesting exploration.
Yeah, we think we're probably going to see mostly sand today. I did see Heterotella, but that's about it so far.
Oh, well, we just started the dive.
I know that we are. We've only been underwater for like 5 minutes, but still.
Yeah. Well, who knows? Who knows?
Yeah, exactly.
So, and whenever, if you need me, just give me a call.
I will, lady. Thank you.
Thank you. We're seeing a lot of these burrowings and the adjacent volcanoes at the bottom of this, at the seafloor here. A lot of this fine-grained sediment. You can see just as the squids touch briefly on the seafloor how quickly that kind of dust cloud forms up into the water column. It gets kicked up. So these sediments can travel really far with even just a low current. As soon as they get kicked up, they can just keep being transported through the water column and potentially having its own, before settling down on the next position. I think that's another sea star off on the left-hand side there.
Yep. Another goniaster.
So we're seeing quite a diverse macrobiology here, even though it's not coral related. We've seen quite a few shrimps, lobsters, fish already, sea stars, brittle stars, and sponges. And we have also some crustaceans, some crabs. There's a smaller crab that's heading straight for the sea star there. Not sure what the goal is. Maybe to get out of the light.
Oh, little crab.
Yep. And maybe what looks like maybe some leftovers of sapulid worm tubes or something alongside it.
Looks like he's got a worm.
Oh, yeah. He's eating
Or an
maybe
amphipod.
something. It seems though the amphipod is it the tube worms or is it just moving?
I think it's just it might be a polychaete
Oh, yeah.
underneath him.
I wonder if it's going to be tried to be ingested.
I don't know. He has to be pretty fast. He's moving pretty quick. I doubt the starfish will be able to catch him.
It's almost like a tease. I'm going to go right near where you spew your guts, but I'm not going to let you get me. Really nice. This is a different color than what we've seen on some of the other ones. These cookie cutters are a bit orangish, but this one seems almost peach colored. It has those platy features on top. Some of the other ones that we've seen have areas where it's almost more porous looking in certain areas, but this one seems to be relatively consistent. This is is that a fish?
I think it's a shrimp. Yeah,
Oh,
it's teeny with the teeny tiny legs. Look how skinny his little legs are.
And he's so translucent.
Too cool.
And it has quite a lump under it. I wonder if that's an egg sac or something.
It could be. Could easily be that.
Or if that's just how it's shaped.
Look how skinny his little legs are.
Can barely see it. Just gliding over the sediment, barely disturbing it. Can we keep the lasers on so we can have rough size averages when we're looking at stuff? Okay.
Mm-hmm.
Yeah.
What's that fuzz
Okay.
ball? Let's snap zoom that guy. Oh.
Is it a coral?
I think so.
Could it be a chrysogorgia?
It could be. I'm surprised. There must be something for it to sit on down here.
What? That is definitely not what we were expecting. It's about 10 centimeters wide.
Mm-hmm.
Wow.
Chrysogorgia, maybe.
I don't know if it's chrysogorgia in particular.
It's got 8 tentacles, it looks like. It's pretty soft. There's a purple polychaete at its base. And, oh, there's a crab and squat lobster in there. And some shrimp. Well, they just blend right in, don't they?
Wow.
I'm
Really
just
large
surprised
polyps.
with that. Yeah, it's
I
definitely.
don't think it's chrysogorgia.
It's
but
definitely a gorgonian.
Yes.
And it might be a Chrysogorgidae.
Yeah.
So
Might
in the
be
same
of that
family.
Yeah.
But it looks awful soft. It doesn't really have those, that like, you know, stiffer stems.
Yeah. So Tina suggests that it looks Acanella-like.
Okay.
See the nodes. I'm not really seeing.
Acanella.
Do you see nodes? Do you see banding? I'm not seeing specific bamboo-like banding, but it does seem to be darker at each of the locations of branching.
Yeah.
But I don't know if that's on the skeleton or if that's more the tissue.
Right.
And those long stalks at the bottom, those brown things could either be dead mangrove roots or seeds, or
Seagrass.
Seagrass also potentially. Oh, and another what looks like
Yep. Anemone.
anemone.
Burrowing anemone, likely.
Oh, looks really interesting. Oh, and it has there's some little brittle stars at the base of it also.
Oh, yeah, look at that. The purple central
Quite
discs.
a few, yeah. I'm seeing 4, maybe 5 right now. What I'm also interested in is those little
Someone's coming out of that hole up ahead. We got a small shrimp or something coming out of that hole.
Oop, and away we go. Probably clearing maybe some of our thrusters.
It's just bioturbated all over the place. There's lots of infauna.
Yep.
This is evidence of infauna. We can't actually see it.
And most of these tunnels could actually be a very complex network under the seafloor, so it could all be connected. Maybe, maybe not, or maybe they're just weaving in between each other. But what a lot of these shrimps do are, as they're building or making these burrows, especially also like crabs, they actually pat. They almost dance around the walls, and they pat along with some sort of mucus that they also develop, and they pat along the sea walls, and they just go all around, all the way up and down. And especially in this mud characteristics, it starts to create almost like a cement-like texture. So it's really hard, in fact, to actually break down that subsurface network system that they have underneath. Unless you're, of course, a very heavy 10,000-pound machine.
Right.
But in the water column, we don't weigh that much. But it's really interesting to see all that, and we can actually see that all. It's usually very well preserved in the rock record if something hasn't come by and really disturbed it all that well, because of that such good cementation that the shrimps and other crustaceans make against the tube walls, and they just kind of like dance along. And my when we were taught this in class, my graduate professor goes around and he actually does the little dance of like of the little shrimp going around. So.
There's somebody in that hole.
Oh, there's a few things in the various holes there.
Yeah.
Oh, Tina said she saw another coral. Not sure where
I did
that
too.
was.
I think it was the same species.
Oh, okay.
That's probably a squat lobster. Yeah, you can see how well formed this burrow is. Right, looks
Multiple
pretty
ones.
stable. Yeah, till we smush it with the ROV thing. So yeah. Sorry, buddy. Give him something to do
another
this
large
afternoon.
brittle star along the seafloor. Ooh, another lobster coming out. Not a squat lobster, but
No, it looks like a snapping shrimp, which would be Alphiidae.
it's a big shrimp.
Yeah, how big is it? How big are we looking here? How big are these holes? Let's get the lasers on them.
He got one look at the ROV and said, "I'm not messing with you." Well, the hole's about just shy of 10 centimeters.
Yeah,
So he's a decent size.
Yeah.
Ooh, that brittle star right at the base of the frame there. Ooh, another anemone.
You did not just do that.
Anemone.
Yeah, that's a burrowing anemone.
Oh, and I think a little shrimp that was just at the base of it and was translucent. Maybe one of the same ones that we saw dancing on the seafloor earlier. I'm going to call it dancing because it was just so elegant. This is quite a muddy territory here. Ooh, we have more brittle stars. These are really long. They have long arms.
They are really long.
We have a dark feature up ahead as well. We can go explore what that is.
Oh, we got something sticking up out of the sand at 3 o'clock. Possible sea pen.
Let it be another coral.
Can't tell till we get on top of it.
Yep.
Excuse me, Mr. Squid. Thank you.
Looks like a coral. It has polyps.
Yeah, let's get a good zoom on that. I think it's a sea pen. I mean, it's got 8 tentacles, doesn't it?
Yeah.
So it's definitely an octocoral.
Yeah. Like a single stalk.
Yeah, I mean, sea pens are.
Or single stalked, yeah.
Yeah, and they are octocorals, so it could easily be that. I will mark it for review.
Yep.
Just to make sure.
Tina says that she thinks it's a sea pen. We're good when you are.
Still lots of stuff down here, even though there's no hard bottom. We've got our sediment dwelling critters. So we've
seen quite a variety of species already.
What is this dark thing? Oh, he was distracted.
I think the
Don't
light.
text and
Yeah.
drive, buddy.
Oh, there he goes again. This looks like maybe some dead stock of something that's left over. Or debris.
I don't know what's happening here. What is that? Looks like a tube worm in the front, and then maybe some debris got caught in the current. Yeah, it looks like human debris to me. That doesn't look. Oh, the black
natural.
thing. Yeah, the black thing does. The thing in front looks like a tube worm.
Yeah.
You can't really get a good. Oh, we've got some kind of amphipod or something on the bottom.
Yeah.
Yeah, that black thing looks like trash. Like plastic bag or something.
And then I think there's another ophiuroid or polychaete around it. No, that's a polychaete, I think. So, looks ugly.
Yeah. Covered in sand. They're just dusted with sand. This one might be pregnant. Looks like it's carrying an egg sac, maybe. This amphipod.
Maybe, and maybe a barnacle at the bottom.
Mm-hmm. Cool, cool.
We can see some skeletal fragments around this muddy area. Usually looks like a lot of pteropod shells. I'm not really seeing any ripple structures, which means there's probably not an intense current systems here. Pilots, are you feeling really any current intensities?
Yeah, as you can see when we touch the bottom, the sand
kind of
pops
walks
up and just kind of hangs out.
Yeah.
Hardly moves at all.
Very slightly.
Nice soft sediment.
Coming from the west? Okay.
Oh, that makes sense.
Coming, yeah, down the shelf.
Mm-hmm.
Ooh, I think that's a skate.
We were seeing finished Raja yesterday. Maybe it's the same kind. It has the same little fins on the tips of its tail.
That was finished Raja Plutonia.
Mm-hmm.
And this one looks maybe a little different. I don't know if it's the same species.
Yeah, similar with the same two tips and the spikes on its down the middle of its back and down its tail.
I'm also interested what's
The
behind
spots.
it. What is that fuzzy thing behind it?
Oh, we might have another one of those. Oh, I don't know what that is.
I think it's a stalk that's covered in schmutz.
Yeah, well, he's really spiny. The other one wasn't nearly as spiny. Now, I don't know if they can change the texture of their skin. I don't know if that's a thing. I know it is an octopus, but I don't know if it's that in rays. But he is very, very spiky.
Yep. Looks like in the back that was a stalk that's now covered also in hydroids. Ooh! A dust cloud on him. So it seems to be about 15 centimeters long, maybe just shy, maybe about 13 centimeters long. The two lasers that you're looking at are 10 centimeters apart, and that helps us identify how large or small something is, the relative size, because things on the deep, our perspective sometimes makes them look either a lot bigger or a lot smaller than they are. So having that frame of reference of those laser points really does help identify things at the bottom of the ocean. Because also we have incredible zoom on these cameras. The specs on these cameras on the ROV are just incredible. So when we zoom in, it might seem huge, but we're actually maybe looking at something only a few centimeters large.
Right. The underworld skate. That's what it was we saw yesterday.
Yep. Don't know if that's the same species.
Yeah, I don't either. It's similar.
Oh, it looks like we have another brittle star in the background, maybe a sea star. We'll have to get closer to get a good identification. Have quite a bit of squid around here. Like we saw yesterday's dive, we were surrounded by squid the entire dive. Oh, we have some little various shrimps around here.
Yep, likely ilex squid. They're almost impossible to tell the species, even with specimen in hand, according to Mike Vecchiam.
Yeah. Yep. And we have a question from Facebook. Sharon Dentinger on Facebook has a question of on the crabs and lobsters that we've been seeing. Do they stay on the ocean floor? How often do they come up? How long are they on land before returning to the water? I do not think these ever approach land. I think they stay on the bottom of the ocean, and they're not usually relative. Oh, it's a sea star, actually. Sorry to interrupt the question. Quite long-armed.
Yeah, so there's land crabs and there's crabs that stay in the water all the time.
Yeah.
So there's certain species of land crabs that come up onto the shore. They're the ones you see in Florida walking across the street sometimes, or in the islands.
Yep.
They usually come up, I believe, to breed and lay eggs, and then they go back into the ocean. These crabs that we're seeing don't ever come to land. They're not a land crab. They are adapted entirely for the bottom of the ocean and the deep sea.
And I don't think they swim very often unless to go quickly to get away from a predator or something where they kind of snap their tail and go off, because it expends a lot of energy. And there's not too much. Yeah, there's swimmerets and something maybe they use.
Yeah, they'll move their legs really quick and kind of bolt off, but they're not.
They don't really swim really up in the water column unless they absolutely have to. So the crabs that you see on land or even in the tidal zones, like the Sally crabs or whatnot, those big reddish ones, those are not the same that live down in the deep ocean. So these guys pretty much stay there. And some lobsters have even modified to, they don't need, because the deep ocean is completely black, they have been evolutionary adapted in that they lost their eyesight. They don't need it because it's all black anyways. They can hardly see. So there's a blind lobster that we saw yesterday, as well as if you joined us just at the beginning of the dive, when we saw bottom, we were able to see another one of those. And they're very palish pink. And they actually lost their eyes just because it was no longer needed. And I guess that was some form of energy that wasn't needed anymore. So they found a way to adapt and save from that. So they have a lot of other sensory organs, such as the antennae and their legs and claws, that help them then search for food on the bottom of the seafloor.
And yesterday we saw one actually actively fishing. All right, so it had a little piece of flesh in its claw, and it was fishing.
Moving it back and forth, hoping to get something, And Mike White says it also depends on the life stage of the life cycle of the crustacean in itself. As a larvae, it could spend more time in the water column before it develops fully into a crab. And I'm sure these guys do molt as well, or no?
Oh yeah.
They molt as well. So
if you look at I think it's planular larvae and plankton, they are the coolest things ever. They look like little flat. When they're larvae, they look like little flat glass, like crabs. It's the craziest thing. I'll post a link to it on my Twitter page, at Deep Sea Nerdy.
Oh, is that another armored shrimp or something? I'm not sure. Let's take a... Oh, whatever. Oh, yeah, it's definitely a shrimp. A squid scared him off. And we're seeing some pockets here and there. More of these bioturbated sections of sediment. Those are those volcanoes that you're seeing. A lot of these crustaceans have adapted to bury then into the sediments, into this finer sediment, instead of using the coral rubble as protection, it has to burrow deep into the sediment in order to possibly escape any predators. And we're seeing quite some squid here. Oh, another anemone. Possibly another little sponge there. Steph, what do you think?
Oh, yeah, Hylanema, likely.
Just a quick snap zoom. Yeah, thank you. Oh, yep, looks like it. But it looks like its stalk is completely buried now.
Yeah, he's really not very tall.
Yep. With like a zoanthid touching the base there, possibly. I'm wondering if all of these tube little structures popping up from the sand are leftover serpulid worm tubes, or...
They could be living
what kind of-
serpulids, too. They
Yeah.
hang out in the sediment like that.
So we're at approximately 635 meters water depth. The temperature is about 6.2 degrees, which is cooler than what we've seen in a lot of other places, which ranged in temperatures from usually about 7 to 9 degrees on the Blake Plateau. So we're still well within all the margins for cold water coral growth as well. And we did see 1 to 2 corals. Oh my god, that's so cute. And
What is
but
that?
we don't have that hard substrate for the coral larvae to really take advantage of and settle on. So we don't really have too much colonization. And besides for those other corals that we've seen, we're not even sure how, why they're there, what they're actually settled on, if they're actually in the sediments, if they were able to find some sort of sedimentary structure that they're latching onto and holding, or if maybe they're burying in the sand. We don't know. We have another fish here. It looks like a cusk eel. No, it has a true fin, tail fin.
Yep, I think it's probably a Lamanema of some kind.
Oh, yeah.
He's awful dusty, though. He's got lots of sand on his head.
I think the lights are confusing him slightly. I don't know where to go.
Yeah, we're scaring him. He can't escape.
What's interesting is that they're usually staying in like a small depression in the sands. As soon as we come up on them, even though they're moving, they're staying within the same little pocket. Oh, we have another quite long-armed brittle star at the base there. Just this vastness of all this muddy sediments that's been bioturbated. So even though we can't always see the macrofauna, it doesn't mean that it's not there. Oh, and it looks like maybe we're coming across another sea pen. If it is a sea pen. Need a closer look. And then off to the right, there's something else that I see that just maybe came off screen. Oh.
This one's different. It looks like he's got those polyps all the way around. Maybe not on the backside, though. The other one didn't have them on the backside. So it's the same species.
Maybe it is. Yeah, the tentacles look very similar to what we saw. And we could be now facing the front of it, so maybe the backside doesn't have any polyps facing in that direction. Nice, one long stalk. The polyps go all the way down to the base into the sediments. I'm not seeing any, besides for that little shrimp off to the side, or amphipod. I'm not seeing really any distinct associates. Thanks, pilot. I think we're good. But I saw something off to the right-hand side that came off screen when we zoomed in. Yep, off to the right-hand side, slightly off screen. There's a little fuzz thing right at 3 o'clock.
Oh, I see it. Maybe one of those
Slightly
corals.
reddish. Maybe a coral with a squat lobster in it. That's maybe the red that we're seeing. Or shrimpy.
Yeah, a little teeny tiny coral.
Yeah, with that squat lobster.
I don't know what coral this is at all. It's growing in. I don't know how it's attaching itself. It's definitely an octocoral, but that's the best I can do. And it's, again, with this purple polychaete at the base.
And it looks like the stem is just digging straight into the soft sediment there, but it could be latched onto a slightly larger grain that we're just not seeing maybe underneath the substrate.
It could also, like, a lot of the sea pens form like this bulb underneath that act as a root.
Yeah.
So it could be doing that.
Yeah, modification of the sea pen and some sort of in the octocoral family, but maybe it modified itself in order to do that.
Select a niche that's not being utilized by any other octocoral except the sea pens.
I think there's a brittle star right at the base of it. Can we get the lasers? The other one was no wider than 10 centimeters, so...
This one might be a little smaller.
Yeah.
It's pretty hard to see in that background. Hey, who just chimed in?
So Tina thinks that this is a form of Chrysogorgia.
Chrysogorgia,
so I think we're in the right family for, or genus for Chrysogorgidae. I'm not sure. Doesn't look similar to a lot of the Chrysogorgids that we actually saw at other locations. But that doesn't mean that it's not related. Those shadows, they look so ominous with the squid.
Yep, so we're going to continue up the slope. Oh, sea spider, stop. 9 o'clock. It is floating in front of the camera. If we can get it.
Ooh.
He looks like he's dangling in the middle of nowhere. There he is.
So they do not form any web-like structure, so he's literally just kind of flowing into the current systems.
That might be something else, too.
Maybe polycnoid?
A pycnogonid?
Pycnogonid.
Yeah, see the sea spider, but...
Oh, okay.
Yeah, he's got...
But usually they don't have abdomens.
Right. But so he's got swimmerettes, so that might be something different, but...
Some really weird shrimp.
Dancing.
Long-legged shrimp.
Yeah, so the longer their legs, the easier it is for them to stay in the water column, right? Because they're dispersing more water. All right.
Wow. That was interesting to see. Thanks, pilot.
As the bioturbation continues, there's something at 9. It might be debris, a dark spot. There's also a few tan brittle stars. There's one at 6 o'clock right off screen. Yeah, I think this is debris, but a quick snap is good. Yeah. Yeah, it's just a piece of seagrass, it looks like. Okay. There's Lamanema at 9 o'clock. These ilex squid are everywhere today, just like yesterday. That one at 9 looks like he's hunting, catching something. Oh. Yeah, so if you're just tuning in, I hope you're having a good Saturday. We are on dive 9. We are off of Key Largo, about 30 nautical miles. We just mapped this area last night. Can we look at whatever's at 9 o'clock? Oh, it's a crab. Oh, yeah, so that's the type of crab. I'm not sure which kind. It's very boxy. It's nice pincers. You can see the modified chelate hand. It's probably good used for picking. So that's his claw. And it's really likely if we look at it from the side, it'll be only really touch on the tips. So it's modified to pick little pieces of food up off the bottom. Or to scavenge. But I honestly don't know what kind of crab it is. He's neat. Probably only about 10 centimeters. Anyway, we started this dive at 640 meters. We're now at 631. We're coming up the slope that we just mapped last night. This is new data. We mapped between our last dive and this morning. So late last night. If you haven't checked us out on social media yet, please do so. We have a couple of pages to share. Let me find them. Here they are. We are on Facebook at Ocean Exploration Research or Facebook at Ocean Exploration Research-Education. We're also on Twitter at Ocean Explorer. You can use the hashtag Okeanos. I actually follow that all day long, so if there's a question you have, it could get read on air. So use the hashtag Okeanos, or on Instagram at NOAA Ocean Exploration. Oh, cool. It's an eel-like animal. He's neat. He's got a wide pectoral fins. Not much of a caudal peduncles, eel-like. I don't know. I'm not good with fish. Do you guys have a name for it? Yeah. He's pretty cool. Oh, and he's about to disappear. Anyone landside have a name for this guy? Yeah, he's about 10. Yeah. There's some shrimp hanging out at the bottom.
Yeah,
Yes.
bye, little fishy.
Yeah, it's a little eel of some kind. I don't know what kind, though.
Some of these burrows are really deep. Unless there's some of those large crustaceans, I'm not sure what would make that.
Yeah, that's the only thing I could. We do have sand tilefish, or tilefish, that do really big. They're pretty big. They're
Yeah.
like a foot and a half long, and they'll make these giant burrows in the sediment. The golden tiles do it. So we might find tilefish out here that would make stuff that big. But otherwise, it's probably the acanthocaris, the blind lobsters.
Yeah.
Just because I've already seen a blind lobster. So I'm just assuming that's what they are. So
Ooh,
we've got
another brittle star there.
some of these small holes. Okay.
Okay. We got some various shrimps we're seeing there. And Tina, that's live in the chat room, that's been communicating very well with us about different corals and sea pens and everything that we've been seeing thus far during this dive. She's actually joining us from Russia, so she's taken the time to wake up pretty much at a completely different, in the middle of the night potentially. So thank you very much for taking the time and dedicating your time to joining us today. So we greatly appreciate it.
It's 6:45 p.m. there.
Oh, okay.
It's not insanely
well,
late.
Not insanely late, but yeah, a little
Like a
grumpy.
frogfish.
Yeah.
Can we zoom in on him? Oh, he's cute.
He has a different color too. It's purple, very small.
Yep.
And you can just tell by the debris that we're seeing kind of float in front of the camera here, there's no true direction where it seems to be flowing towards.
Oh my gosh, that fish is so tiny.
Yeah, can we get the lasers on when we zoom back out?
Oh, he's so cute.
Probably less than a centimeter big.
Yeah, he's a teeny tiny fish. So it's a type of frogfish. So he's going to have those modified legs underneath so he can kind of walk around. He's itty bitty.
Maybe they're a different color when they're juveniles versus when they're older.
Yep,
I haven't
totally.
seen too many frogfish of this color.
Totally possible. And a lot of fish can change color.
Yep.
Depending on the species. He's too cute.
Can we get lasers? Oh yeah, maybe just about 2 centimeters big.
Tiny guy.
Yeah.
I can't believe we can see that. Just for context, if you guys look on feed 2, you can actually see our Sirius filming D2, which is the main ROV.
It's a fantastic view.
Yep, and D2 is 9 feet tall, so it gives you an idea of how big this ROV is that you're seeing from the Sirius feed. So the fact that we can film a 2-centimeter fish from this 9-foot tall, 10,000-pound machine is amazing.
Absolutely incredible. Just goes to show how much high quality mechanics goes into building some of these machines.
It makes a huge difference for like on the research end because
absolutely,
I have to look at thousands of photographs. Every time I go on a cruise, we take thousands of bottom photographs, and I have to look at every single one, and I have to determine the percent cover using this computer program. If I can't make out what it is, then that data is lost, right? So that data ceases to be valuable.
Yeah.
So when you have high quality imagery and high quality photographs, and you can make out the teeny tiny things that don't really seem to matter to, people don't think they matter, but they do. They're food for other, they biodegrade the bottom. They
can then
cycled carbon.
Yeah, and you can't tell the different even species of coral that there are, because we need to look at the polyps, and even at that point you have to look then at the septa that are in the polyps. And it's incredible to determine what kind of species and what variety we're looking at here. So in order to have that really good quality imagery is extremely important.
Right, like the Elizella elongata we saw yesterday. Like, I'm used to seeing that as a white stick. I'm not used to being able to zoom into it and then tell where the polyps are. Like, I know that as a white stick.
Yeah.
So, like, I saw it and I was like, I don't know what this is, because I could actually see what it was.
Yeah.
Whereas what I'm used to looking at is just a white stick.
Yeah.
Right? So it makes a huge difference when you have high quality video and photographs.
And most of the time, you can't identify these things unless you have it or a specimen in your hand. And the fact that we get to videotape this and take images of organisms in their environments, and we get to see how they behave.
Looks like another armored sea robin. That is Tragliidae.
Those modified fins it's like a fish crab.
Ooh,
that was shaking about. Probably those little antennae-looking things jutting out from the front could be also modified barbells to help them sense things even wider than it is, cover more ground.
I would think so, because they're coming from that under the chin area, which is where the barbels are usually located, and it looks like he's almost combing the bottom.
Yeah.
Right. Like combing the desert.
And you see some other barbells under him. God, those legs.
It's so neat. He just kind of tiptoes. Tiptoe, tiptoe.
And again, the goal at the bottom of the seafloor, given how sparse food can be, is to move with as minimal effort as possible, to expend the least amount of energy that's needed. Because you don't know when the next food source will come around, so you kind of have to minimalize how much of that energy you're expending to get to the next food source.
Looks like we've got a shrimp dead center.
Yep. And a squid hanging out at the bottom there. Oh, just taking off. And some of those darker leaf-looking objects on the seafloor that we've been seeing sporadically, not continuously, but in various places, they could be decaying mangrove roots or seeds that are coming off. We know that the entire keys are lined with mangroves that are very important for the overall ecosystem on the coastal side, and preventing and trapping a lot of those incoming storm waves that are caused by hurricanes. Those could be those seeds that maybe just don't make it to another location to start growing, or there could be some Thalassia seagrass that's been uprooted and and cascaded over the shelf edge, and while decaying, they become slightly more dense and they fall to the seafloor. Saw quite a few of those long arm brittle stars there. And I mean, I'm really curious as to what could be making these really large depressions. It could be the blind sea lobster, but I don't know.
If we can find an occupant, we can figure it out.
Yeah.
It looks like there's, active holes here. So if we look, well, that one maybe not, but some of them are filled in, some of them aren't. Can you guys pan up just so I can see ahead of us for a few seconds? Okay. So, yeah, we have a very slight slope. It's hardly anything. On the multi-beam, it looks a lot steeper.
Could be exaggerated.
Oh, here's somebody in a hole. If you look at 6 o'clock.
Looks like a crab.
Yep, could be. Oh, damn. He went away. Yeah, we freaked him out.
Yeah.
Yeah, he's way deep in there.
Just goes to show how intricate this network system is.
Yeah, I mean,
right
that could
under
be
the
his back
surface.
door over there, or his side door, or his neighbor. It's a neighbor's door. Could be the back door back there.
Yeah. All of these holes
Yep.
could be somehow related to that same one or just woven around each other. Another one of those sea robins. Oh, what's back there? What is that? Wondering what that is. Get a quick snap zoom. Is that a squat lobster? Is he alive? Well, it
He's
looks
got
alive.
really long arms.
Yeah.
Might just be a shed, right?
Oh, like a molting leftovers?
Yeah, the molts are weird because they look like they're real. They look like a living. Oh no, he's alive. He's alive.
The
You can see
antennae
his
are moving.
antenna. Yeah, he's got really long arms.
He's quite large for something. Normally, they have associated corals or sponges or something. So
He's got lots of little shrimp on him, and he's pretty cool.
Quite spiny.
Definitely a squat lobster, which would be Galathioidea.
I think he's missing a leg. One of them looks stunted. Oh, it might be on the move a little bit, looking out on that the shrimp getting maybe too close for comfort.
Squid.
Squid, sorry. What did I say?
Shrimp.
Oh, darn.
I mean, to be fair, we've been staring at marine life for like... Oh, look, you can see his tentacle hanging out.
Yeah.
Let's see. Yeah, so Galathea.
Well, there seems to be, I don't know if that's a hole or if that's just a dark object in the background there at the top.
There's another Laminema in that pit down there.
Oh, yeah. Oh, looks like some shrimp. Really long antenna, quite clear except for the tail. I don't know if it's meant to be vertical. Maybe he's hanging
Oh,
out up in
fun.
the water column.
Yeah, he is probably pelagic. He's really clear.
Yeah.
He's pretty good at swimming. So he probably hangs out in the water column most of the time. You see his swimmerettes.
And the positions of all of his legs and arms.
Yeah, he's really skinny.
Yep.
So he made himself real thin.
Probably to help move around in the water column.
I guess he really doesn't have an up. It's kind of whichever direction I'm facing, I'll take it. Let's see what happens when he hits the bottom. Let's see if he becomes a bottom dweller. There's another little shrimp on the bottom over there.
Yeah, and a brittle star in the background. La da da dee da. Da, da. This is very serene. Oh, and you can see, I mean, if he's pointing towards a certain angle,
Yeah,
like you can almost can't see him anymore.
Yeah, he's totally invisible.
Tina says, Sir Guestiday? Sir Guestiday?
It looks like a rat.
I don't know, I don't quite see it.
I don't know what she was looking at. Maybe she's on a delay. Hang on. Oh, yeah. Sir Guestiday. That's a prawn. Since the Jurassic. Pretty cool.
Nice. They are mostly pelagic species, I thought I read.
Awesome, Tina. Thank you.
Yeah.
That will be going in my notes.
I don't think there's a favored orientation.
Yeah, he seems just kind of
Wafting
go whatever.
about.
Yeah, go whatever direction the current takes him.
He's just barely skimming the seafloor, not really touching it. Never really truly landing.
So shiny. It looks almost like he's made of plastic. Oops.
Oh.
All right, that's counterproductive.
I've fallen and I can't get up.
Yeah, right. You gonna take a nap?
I don't know if that's a good idea, buddy. Wonder what that dark thing is. Looks like, again, more dead, decaying mangrove seeds, possibly some thalassia seagrass that has decayed and come down to the seafloor. In the distance, there seems to be some darker things sticking up out of the sediment. We can get maybe a look at that. Not quite sure what it is. Yeah, coming in den.
There's an, yeah.
Yeah. Oh, yeah.
Oh, it's
There's
like,
a
it's that
crab.
crab. It's that little white crab. I don't know, with a fine pincher.
Yeah.
The giant pincher claw.
Nope, scared him back into the hole. And then slightly above that, is that a sponge? It looks like a sponge. Almost approaching dead center now.
Oh, it might be a. Oh, can we zoom in on that? That might be a collectible. Right behind that. Yeah. Because I believe that is a carnivorous sponge. Get out of here, silly squid! Move. Yeah, we probably need to grab this. It is a carnivorous sponge. Oh, how cool. Chris, I hope you're on the line. You need to call me. We need to collect this. These are super rare. I've only ever seen one before, and it was along this area closer to the Pulley Ridge. This is a carnivorous sponge. The first time it was described, I don't know the species. The first time one of these was described, the guy called it an alien antenna. He had no idea what it was.
Well,
But
Scott's calling it related to the Cladorizid family again.
Yep.
Looks like it has a polychaete on it as well.
Yep.
So it looks really cool. It looks like the lollipop sponges that we've seen, but now without any of those hairs.
This one, if we can keep it intact, guys, that would be amazing. So your best bet is probably to come in on the bottom. Ideally, yes. Yes. Hi, Chris.
Hi.
Has your mind exploded?
Kim, how are you?
Good.
Good.
Are you excited?
Good. Oh my God. Oh my God. And I have never, described any of these species. Chile has recently. And wow, for the habitat, I know it's easy to survive as a sponge here. But definitely carnivore sponge, the family Cladorhisidae, and different from the ones yesterday.
Oh, yeah.
Oh, yeah.
Like Cladorhisa, but I cannot give 100% identification.
Yeah.
Now, these round bubble structures that we see are reproductive structures.
The round bulbs are reproductive structures?
Yes. The
Awesome.
round balls are reproductive structures. Yes. You don't have them all the time, but when they are producing eggs and sperm, these are hermaphrodite sponges. And I will be researching a little bit more to tell you exactly, what they are. But I know that they contain reproductive material.
Oh, nice. Scott is calling them chondrocladia.
Chondrocladia?
Does that sound familiar, Chris?
Chondrocladia.
Chondrocladia?
It could, yes. Yes, it could be. It sounds familiar, but, since I'm not really an expert on this group, I will look a little bit at the literature. I started to look yesterday when we got the tons of carnivore sponges. And this is really unique. This never reported for this region and maybe even for the Caribbean. So, and the wider Caribbean. So really need encounter.
Awesome.
Wow.
Okay.
Great find. I'm glad we came across it.
She was very excited yesterday. I sent a couple of the pictures. She was like really, really moved by this discovery that you guys are doing with the carnivore sponges.
Awesome.
Excellent.
Thank you.
I'll keep in the line. I will update you in anything else that I can do.
Okay. Thank you.
Thank you.
We got to clear this sediment. before we can see what we're doing. A squid wrecking ball.
No problem.
It's like he's Pigpen from the Peanuts. The ball of dust following behind him.
As long as we don't lose the sponge.
Yeah, it's right there. That's good.
You can see how fine grained this area is. You can see the imprints of the ROV in the mud.
Right. It's going to be very fragile. Just keep that in mind.
Talk to Shannon. Might be worth trying to dig under it a little bit. Ooh. Oh, there it goes. Or, oh, not yet. There you go. Yep. Oh, I think I see it. Oh, that's the ophiuroid. think it got sucked up. Yep. So Chris does think that it's agree with Scott in that it's a chondrocladia, the number one species in the genus, once described by Schmidt in 1880 from the Great Antilles, and this could very well be undescribed and never reported in this region. So thanks, pilots.
Is that a squid at 9 on the bottom?
I don't know. It's about to come off screen. I think that's, yeah,
Squid
ceviche.
bits.
Ceviche squid.
Oops. It's food for the shrimps, or crabs we're seeing. Yeah, did you? Oh, we got a crinoid. Yay! A 10 armed. Long tips. So awesome. So that's the first crinoid we've seen in a little while. There was none yesterday. It's a 10-armed crinoid with the long tendrily tips. I had a name for it last week and I'm blanking. Awesome.
So we saw our first crinoid. We didn't see any yesterday at yesterday's dive when we were closer up on the Miami Terrace. So that was an interesting note. We saw some variety of sponges and a few corals, but not a single crinoid. I wonder why that was.
I have no idea. The, I am looking for the name still, so
Well,
hunting it down.
well, we have a question from Twitter from
Octopus.
Ooh, we'll get to your question in a second.
So it looks like a bobtail squid. So the little tiny round eggs we saw yesterday was
That's
probably
from them.
from one of these types. He looks like a little squeaker toy.
Yeah, I was about to say it looks so cute. Really small. Can we get the lasers? Maybe about 4 centimeters. Ooh, getting a little cloudy. Don't go anywhere.
He's too cute.
Oh, that looks just adorable. The eyes almost take up the entire body.
Yeah, he's so cute.
Has Pixar made an animated show about this one yet?
I don't know. They need to, though.
Well, they need to, yeah.
I mean, they have 2 whole movies dedicated to you know, like Nemo, right?
Yeah.
Add a bobtail squid in there somewhere.
Have you seen the little animated spider one? Like little Charlie the spider?
No.
Where he makes you all friendly and
Oh,
cute?
yeah.
They should make one of this one.
Look how cute he is.
Looks like the ocular lenses go pretty down deep into his body.
Yeah, because it's see-through, right? So you can see how big his little eyeballs are. Oop, squish down, don't get eaten.
I'd like to have a side view of him, but I don't want to kick up more sand.
It's cool, he's got these like light blue color. You can see his little suckers
Yep.
on his arms that he's got tucked.
Staying very still.
He's too cute.
We're good when you are. Oh. It's so cute. No more than about 3 centimeters.
Yeah, he's tiny.
Maybe a little more, maybe about 4. Still really tiny if you take that into perspective. Again, we're talking in centimeters here. Bye, buddy. So to go back to that question now that we had on Twitter from the good doctor. I'm surprised that some of these small fish out in the open aren't being picked out by the much larger squid that are seen everywhere. Is there something about the fish that makes them unpalatable?
I don't know.
Yeah.
That's a fish question. Or a Mike Vecchione question.
Yeah.
If Mike Vecchione were on the line, we'd be able to get a much.
Maybe the squid just aren't hungry?
Yeah, so they could be they could be migrating to breed, in which case they're not really interested in eating that much. That's a possibility. Because they all seem to be heading north.
In a single direction.
Hi, did somebody chime in?
Oh, I think someone's just logging on to the conference call.
I was hoping it was someone to chime in and answer for us. And I was looking up, I was looking this up for a minute to see if I could find a better answer than what I had, which was basically I have no idea. The sea robins are edible by humans.
Okay.
So they're probably difficult to get through, but these, like as far as the tissue on their backs, right, to get through that if they were to get caught, because they're armored, but they have these. I mean, these squid have really nasty beaks, so I can't imagine they can't get through that that tissue to eat them. I really just don't know. It looks like
Mike agrees that they're pretty good eating.
That looks like somebody's tail. Stuck in a hole or severed. I can't really tell. Put his head in the hole?
Orange, just in the sand. He's like, if I can't see you, you can't see me.
If it fits, it sits.
Yeah, not sure. Yeah. Can't really make out what it is. Maybe some eel-oriented fish.
Hey, Okeanos.
Hi.
Hi. Who do we have on the line?
Hey, Mike
Mike
Becky,
Becky,
I'm here.
yeah. Okay, so you're
Hi.
here to answer the squid question. I knew it.
I just tuned into the video and I saw a bobtail sitting there, and then I heard you asking questions about squids and interactions with fishes and stuff. So I thought maybe I'd call it. Actually, my wife told me to call in.
Yes, please. Thank you.
Thank you.
So the question was,
what's
did you
the
hear the
question?
question? It says, I'm surprised that some of these small fish out in the open aren't being picked up by the much larger squid that are everywhere. Is there something about the fish that makes them unpalatable?
It depends on first of all, whether the squids are hunting or not, and secondly, whether the little fishes are moving. There's a lot going on there that makes that question difficult to answer. But I think somebody was asking whether they might be interested in something else, and it's the time of year when ilex spawns down in that area. So it's possible that they're getting together and getting ready to have a mating aggregation.
In which
I haven't
case,
seen any evidence of that yet.
they wouldn't be eating if they were in the middle of doing that. If they were on their way to have a mating aggregation, they probably wouldn't be eating much or hunting much.
Well, I wouldn't say that.
Okay.
But, squids are very active, and it's possible that they would do both at the same time.
Oh, great.
Um,
They need a lot of energy, right?
I don't have a real solid answer for you about that question.
Okay.
I can tell you that in the late fall and into the winter, the ilex actually made some spawns down in the area off of Florida.
Okay. And that's probably where these guys are headed.
We've seen spawning aggregations of ilex from Okeanos before down off the Keys.
Oh, nice. Well, thank you very much for paying attention on your Saturday and chiming in to help us, because I had no idea what the answer was. So
I just tuned in for a minute to see what was going on, and I was more interested in that bobtail.
yeah, it was cool.
Very unusual color.
Yeah, it was really pretty. It looked like a little squeaker toy.
I didn't see how big it was, but I wonder if it was a recent hatchling.
Oh,
it might have
maybe.
been. It was only about 2 to 3 centimeters long.
They hatch pretty big, so it's possible, particularly since we saw what looked like it might be bobtail squid eggs had been hatched out.
Yesterday, yeah.
That's in the last dive, yeah. So it's possible that that's a pretty recent hatchling of one of the bobtails.
Wow.
Awesome. He was too cute.
Yeah. Out into the brand new world.
Thank you so
Thank
much.
you.
They're never too cute.
Oh, yeah.
Yeah.
Well, that's true.
Well, you should stay tuned in. We don't know what we'll come across down here.
Keep us on in the background.
I have other assignments to do today.
Oh, please. We're more important. All right. Well, thanks for chiming in.
Well, yeah, if you got other questions, you know where I am.
I know. I'll yell out for you, and you'll call in. Thanks.
All right.
All right. Bye.
Bye.
Just like that, we have our answer. We love
I
it when
know.
other scientists onshore can call in and help us identify certain things, or maybe talk about certain anecdotes that we didn't catch. Like yesterday, Chris Ma identified that part of the sea star around it was actually relatively clean rock compared to the other portions of the rock that we were looking at. So maybe it was feeding on whatever hydrozoans or things that were clustered on it. So these are all things that our eyes aren't tuned in and trained for when it comes to a lot of maybe the associating biology or the behaviors of certain animals. So we really appreciate anyone that can call in or chime in on the chat room to let us know and inform us about what we're seeing and what's going on down here. Because you never know what we're going to come across.
Right, like this baby Lemonema.
Yeah.
Little fish.
Really small. And translucent, I wonder if it's a juvenile that will grow darker as it grows.
I think so. He looks like a baby. And it doesn't look very
Maybe it's hatchling
mature.
season.
I think we're freaking him out. He can't escape the light.
But yet another staying put in another depressionary feature in the sediments. So maybe they're not the designators or the creators of these features, but I think they definitely maybe live in them and take advantage and contribute maybe towards the depression in itself.
Xenometricolumnaris. That was the crinoid we saw that has taken me like 10 minutes to find the name of.
Well, determination.
Yep. It's driving me crazy.
Was that the crinoid we potentially sucked up as well?
I think we sucked up a brittle star.
Oh, okay.
Yeah.
Ooh, another shrimp hanging out there. You can see, kind of make out its really long antenna. Nice. You can barely see it just along those red sections there. Seems to have buried itself. This was maybe what those shrimp that we were looking at yesterday were doing. This was potentially the end goal there.
Yeah. He's really buried himself in the sand. Hiding from those big squid.
Potentially. I think he's done quite a successful job there. Yeah, we're good to go. So we have another question on Twitter from Helen. Why are the squid that are passing by sometimes bump into the ground? Well, that could be due to the confusion from the lights in the ROV, that sometimes they're blinded or they're not sure what to do, so they aim to go downwards. So they maybe think that they're too close to the surface where there is light, so they aim to go further down, deeper into the ocean and bump then into the seafloor because they think there's a lot more space to swim down than prior. Than if they were at the surface of the ocean. So the RV does cause a little bit of confusion with the overall light systems that it has, but since the deep ocean is relatively dark and black and you can't see anything, but we need these lights in order to see ourselves and to navigate where we're going and identify certain organisms. So for us, they're quite important. And besides for some minor confusions here and there, I don't think we're causing too much of a interruption. As soon as we move on, they'll go back to their normal lives, swimming about.
Right, so they do migrate from the bottom to the surface.
Yeah.
So if they see light, their natural instincts is to escape that and go down to do it, right? So they just kind of barrel into the sand by accident.
Yep. What is that dark object off to the 3 o'clock there that's coming off screen?
Probably some detrital material.
Possibly.
Sargassum. Oh, there's a big burrow on the side. Yeah, it looks like algae. Sargasso.
Oh, yeah.
Sargassum. Floated down from the surface. Still pretty living.
Yep.
It's a brown algae.
So I came across a recent documentary that talked a lot about these sargassums, that fish tend to brood underneath it and attach their eggs and larvae to this entire maybe patch of sargassum that then becomes so densely packed and heavy that it ends up sinking down into the deep ocean, which then provides protection for these larvae until they're ready to hatch and escape then into the ocean. So it's an advantage for those fish to not get eaten, and considering that the larvae are filled with really good nutrients for all the other animals, it was a way for them to protect themselves with it by implanting it on a sargassum
Looks like a
patch.
sponge, maybe?
Yeah, I think so. Another one of those hylonemas that we've been seeing.
Oh, possibly. It's just a tiny baby, so it hasn't
Maybe.
grown not long enough.
Or
Spicules.
the spicules have been buried by the sediment.
Yeah, in the drift.
Yeah.
Is there anything going on in this divot up here?
Not seeing too much.
Might be a filled-in burrow. It's pretty big. Abandoned burrow. And then we got a little shrimp.
I don't think a shrimp created all that.
No.
Yep. And we're grazing along the seafloor. If someone at home is just starting to tune in, we're on the southeastern end of the United States, down by Florida, closer adjacent to the Keys area now. Yesterday we dove on the Miami Terrace, which is that continental shelf that's right off the break of Miami. Now we're heading a little further south here. We're making our way down to Key West and dive in some of those areas. Oh, I think that's another sea robin, if I'm not mistaken. The front view of it. Quite nice. You can see his modified fins. Those look like a lot of crab legs that are underneath him, where he's actually capable of walking along the seafloor. And then those, what looks like extended barbells there with spikes on it, are probably used to sense anything moving right underneath the surface of the sediments there. Probably sensory related. And you can see now how he just used his modified fins to walk along the seafloor. And he's an armored sea robin. You can tell he has quite a few spikes around him. Maybe he's taking a defensive stance now, like in case you try to nibble on me. I have some power. You can see his, I think his mouth was just opening up there. Oh. Not sure if he was trying to suck something in intentionally. Seems to have a really intense snout-like feature with two prolonged ends there. Thanks, pilot. Oh, it looks like we have some debris out on the farther end there. Oh, maybe some piece of plastic or whatnot or rubber with a squat lobster on them. Taking advantage. Sitting a little higher in the water column there. Maybe it looks like, yeah, some piece of rubber from a cable or something. Oh, we have another squat lobster there. So I'm guessing these are two different species just by the sheer color and the shape of what they are. Normally they are associated with some form of standing biota, either, ooh, is that a sea cucumber? Straight ahead. they're associated with some form of standing substrate of like corals or sponges. Usually they try to end up higher in the water column and associate themselves with it. Oh, this does look like a sea cucumber. Ooh, I'm not sure what genus or species this is. If anyone on the chat room might know, it looks like it has some spikes along the side as well as along the edges, probably due to deter any predators from trying to take a little bite out of it. Not exactly the most pleasant of feelings. Looks like he has some long antenna on both ends, so I'm not entirely sure which side would be the front or the back end. If there is a front or a back end on them, you can see there's some large antennae there in the front. Maybe to sense some stuff. He's moving ever so slowly, but he's moving. Oh, got a little cloud of some mud dust coming up. But you can see that there is in the back of the potential sea cucumber, we do have also the same kind of antenna that are possibly, well, what I'm assuming is the front end. But it could very well be either or. And Tina on the chat room says that they're often also crunchy because they have potentially some skeletal elements within this its system there. So does that still does that make it still qualify as a sea cucumber? I'm not sure. Or maybe a larger sea slug. Holothurian, yeah, something of the sorts. Can we get the lasers to see roughly how large it is? Oh, we're getting another good look at it after the sediment settle down. Seems somewhat active. You can see those large spikes along the sides there. Definitely something that would deter, I'm guessing, a hungry predator to try to snack on this guy. It seems he is moving. I'm not seeing any particular kind of tubular feet or anything underneath it, which sea cucumbers don't always have. Some species have them, not all of them. Could be also a form of a sea slug that comes by, which also don't really have tubular feet. I'm seeing some micro forms of maybe something to help it, like pinnulae that help it move across the seafloor. Ooh, moving. I'm guessing maybe the area that I've been calling the front end is the front end. Potentially, since it seems to be moving in that direction. But again, sea cucumbers can technically change which direction they call the front end versus the back end. So, really nice to see. I think we're good to move on whenever you guys are. Can we get the lasers one more time, just in case? Ooh, he seems about 20, more than 20 centimeters long, maybe about 22 centimeters long. Thanks, pilot. If you're just tuning in here, we're on the seafloor towards the adjacent to the Key Largo on the deep ocean there. We're scaling up a very gradual slope. It seems like just a few degrees, and we're coming across a very fine, muddy carbonate sand in this area with a few skeletal elements, so a few coarse grains here. Ooh, is this a tripod fish? Get a... That would be cool if it was a tripod fish. It is a tripod fish! Oh, they look so cool. You can see their modified fins in the front are meant for sensory applications, and especially those ones that come directly up above it. It almost has like antennae, and that's meant also for some sensory applications. And you can see the pectoral fins and the tail fin. It's called a tripod fish because it uses those modified fins to just stand on the seafloor, and it has exactly 3 points where it touches the seafloor. Those two pectoral fins midway through the body, and then also the tail fin that touches down. So really interesting. We haven't seen one yet on any of these dives. So, and they come in various colors, I think, depending on the certain regions that they are. So really interesting to see. You can kind of see those antennae at the top, the really long ones that come out. They almost look like a fan-like structure. And it uses all of these for sensory applications in order to sense anything in the water column or around it, maybe to make a quick getaway or a quick snack. And they can be relatively quick. They spend a lot of time stationary, hanging out on the seafloor, as you see now, due to the fact that, as we discussed earlier, there isn't always a plentiful amount of food at the seafloor, so we need to save a lot of that energy, and this allowing him to stay stationary and sitting on the seafloor like this exactly allows him to do that and save on that energy, while using a lot of those sensories organs to help him better estimate, or help it better estimate where the next potential meal might come from. Really nice looking. Can we get the lasers out to see size average? Ooh, he seems to be just about 10, maybe slightly more than 10 centimeters long. Really nice find. Thanks, pilot. I'm not sure of the entire species, but I think we're looking at Bathypteros. I'm sorry if I'm butchering those scientific names. Was that tripod fish the scientific name of that tripod fish? Looking up, we see more brittle stars scattered about the seafloor there. Again, this is very fine-grained sediment. This is different than what we've been seeing on a lot of other dives here. Doesn't look like we have a hard substrate underneath, at least not anywhere near the subsurface. So we know that there's quite a bit of sediment that's draped along this seafloor, rather than maybe before we saw some sediments that were only draping barely. Oh, can we get a zoom in possibly on that coral that's there? That's been draped along some hard substrates, whereas here it seems like it seems to be a very dense package of soft sediment in this area before we reach any hard bottom substrate. And you can kind of tell that by looking at all of these sandy volcano-looking plumes in the background of the area here, and that's due to the bioturbation, where a lot of shrimps and other crustaceans and possibly fish burrow down deep, and when they're creating these intricate burrow networks, they waft up a lot of this sediment, and it piles up and creates these volcano-looking little mountains piles on the seafloor. We seem to also have another armored shrimp that's just off camera, pretty close by. And it looks like we have another one of these corals that we've been seeing. So we've seen now 3 specimens of this coral, and they all seem to be the same species. We'll let the sediment waft away a little bit. I'm not seeing an associated squat lobster here, but this seems to be of the chrysogorgid family. Tina online is suggesting a bamboo coral, but I'm not really seeing those distinctive band structures that we've been that we identify a lot of bamboo corals with. Let's let the sediment kind of pass by a little bit before we can appropriately say that. It seems though like some of the tissue where the branches are kind of forming out from these nodules are it seems that the tissue might be slightly denser, giving it the illusion maybe those bands are there. But I'm not entirely sure that we can really see that, that we can conclusively say it is a bamboo coral or not. So hopefully we'll come across another one, and then maybe Scott can chime in, although he said it sure looks like an Acanella coral. So definitely something intriguing that we're coming across. Seems that I had just talked about the fact that there's not any hard substrate in sight. So maybe this is one that is very similar to the sea pen, where it creates that bulb that forms just below the substrate, and it kind of creates almost its own hard substrate and sucks in a lot of seawater, which makes it dense and holds it then in place as then the coral can grow. But we haven't seen it larger than about 10 centimeters at most in size. And these are another one. We just saw one earlier about 20 minutes ago of these. We're not entirely sure what it is. It looks like a sea spider, but I think it might be a pelagic shrimp or something along those lines. Can't really make out what all the limbs are. I'm not seeing any active swimming going on, more as drifting in the current systems. Some sort of isopod. Maybe. Oh. Looks like it can touch on the seafloor and can walk. Looks pretty stable, grabbing onto the seafloor there. Yeah, some form of isopod, where sea spiders don't usually have an abdomen associated with it, and this looks like it does. The one we saw before, oh no, the sediment. The one we saw before was drifting through the current systems and didn't touch down, as far as at least we could see. This one has touched down, so maybe they do spend some time they're more pelagic in that they spend time more in the water column than on the seafloor. Maybe their long limbs help them regulate how far up or down they're going in the water column. Scott is calling it a monopsid idopod. So thanks for that, Scott. seems to be able to hold its ground on the sediment despite it being, maybe he meant isopod, looking so fragile. Thanks, pilot. Isopod,
Hey,
yeah.
Okeanos, Mike Vecchione again.
Hi, Mike.
Hey, I'm not on the chat line, so I thought I'd call to let you know what that spidery-looking thing was.
Absolutely.
That's a munopsid isopod.
A munoptid isopod. Interesting. And how are they similar or different from the sea spiders that we saw a couple dives ago?
Oh, it's a completely different group.
Oh, okay.
You'll see sea spiders swim once in a while, very, very rarely. But those are pycnogonids, and the munopsids are isopods, which is a crustacean group. And you very often see them swimming.
Nice. Do they normally live within the water column, or do they kind of more use it to transport themselves until they get to the next favorable spot where they want to settle on the substrate?
I think it's the second option, but there's a lot of species, and it's outside of my comfort area. So I'm just telling you what I know about them. So I do know that they go down onto the bottom, but we see them up in the water column pretty often, and it's a very diverse group.
Okay.
Particularly the benthic species.
Okay, and to determine their diversities, you probably need a sample in hand. I mean, I'm sure despite the incredible video zoom in that we have here, it seems to be quite difficult to be able to tell them apart.
Well, you could send a frame grab to Karen Osborne at the Smithsonian, because it's one of the groups that she works on.
Okay.
But like I said, I think it's a pretty diverse group, and I suspect that you probably need to get a specimen in hand so that you can count the little hairs on the legs.
Oh, geez. Okay, well, we'll definitely reach out to her. Maybe she can tune in and call in and talk more about those as well. So thank you for that information.
Okay, just wanted to make clear that it wasn't a
sea spider.
pycnogonid.
Yeah.
Was, sometimes we call them water walkers. It's
water walkers. Oh, interesting. Great. Well, thank you. That was great information.
Okay, I'm going to hang up now.
All right. Thanks, Mike.
Bye.
Bye. And Scott is chiming in on the chat room that these monopsid isopods are more closely related to the bathynomids that we saw one yesterday, these bathynomids isopods. So those are the Bathynomus gigantus, the large isopod roly-poly looking creatures that we did see one yesterday than they are to actually the sea spider. So they're more closely related to that giant cockroach looking thing we saw yesterday than actual sea spider. So really interesting tidbit. Even though they can look so similar, and I mean, I don't have trained eyes for these different types of characterizations, but it looked very similar to the sea spider of what we were looking at. But it's great that we have scientists on shore that can really help us and tune in and let us know what we're dealing with here. Ooh, and you can see here that the squat lobster that's hanging out here, he just kind of went totally flat against the subsurface when maybe a squid floated by. So maybe he's taking a defensive stance and saying, nope, you don't want to eat me, I'm hiding, everything's good. It looks like a shrimp or another kind of little lobster. Squat lobster is resting on top. Maybe can't really make out what it is, but it seems to be hitching a ride. Really interesting to see it. Maybe it looks like a shrimp. I can't tell if that part on the left looks like it's claws or if that's more of the tail portion that's there. Yeah.
Yeah, that's a shrimp on his back, I think. A broken
little
back shrimp?
hitchhiker, yep.
I think the other one had one too. I wonder if they're commensal.
Did it? I don't know. But Scott also said, as I was talking about that coral that we saw earlier, looks to be like the Acanella. Scott chimed in that saying that the best way to visualize the roots of that and why it can actually stabilize itself within the sediment is to picture a candle wax dripping from the base of the candle. It forms a calcium carbonate skeleton that kind of makes its own little root systems and what are called rivulets through the sediment. So it's not quite bulbous like the sea pens create, but they actually form their own little root systems. Very similar, I guess, what you can describe also from the acrinoid root feet-like structures that they kind of bury into the sediments and they grow there. So if you can picture candle wax kind of dripping through and it burrows kind of deep into the sediments, we can see that.
Here's another one of those.
Is that a... Oh, another one of the sponges. Carnivorous sponges.
Carnivorous sponges. That's awesome. So we collected one of these maybe half hour ago. We think it might be either new to this area or entirely undescribed. Cool. Is that what do we got here? Oh, a burrowing anemone. So he actually lives in a tube. And the tube is underneath the sediment. And if we were to freak him out a little bit, he would pop in, hide himself in there. Pretty cool. You can see his mouth is right at the center of all of his tentacles. Oh, this is not a Cereanthid. What kind is it, Scott? Edwardicidae? Edwardicidae? I wonder where that name came from. It's a true sea anemone. It's a burrowing anemone, but not a tube anemone. Okay, thank you. Edwardicidae. Edwardicity. All right, it's lunchtime, so everybody is kind of changing shifts here. Still have lots of bioturbation in this area. It looks like this is an active burrow at 6 o'clock. Actually, there's a couple of them right here, some at 3. They look like they've been actively dug out versus the rounded, filled-in ones, which I would consider inactive. I think a lot of these small holes are caused by that crab, and I'm not sure the idea of the crab, but it's a small white crab, looks similar to Chaseon with that boxy back, and has a very large pincer-like chelate claw. They're pretty, we rarely see them, but I have seen a few, and they have been in these little holes. So my guess is they are the ones building this these smaller holes. The larger holes, I'm not quite sure. They could be acanthocaris holes, which would be the blind lobster. They're pretty common in building these holes with a front and back door. The larger ones. We saw some acanthocaris yesterday. I saw one earlier in the dive, which makes me think that's what they are causing. I mean, are the ones building these holes. Ah, we got another sea pen. Yeah, we've got, this is the same sea pen we had been seeing, since the beginning of this dive. It's, all the polyps are on one side of the stalk in the center. You can see the really distinct 8-armed, 8-tentacled polyp. They're quite large. I do not know the name of those sea pens. This is Xenometra, likely the crinoid, a 10-armed crinoid with the thin tips. So this is, we've seen a few of these this dive. Oh, and it got dusted out. I see some worm tubes at 4 o'clock, just at the edge of the screen. We've got a crashing squid. Looks like a tiny Menopsis shrimp at 4. Nope. He swims off. I guess we can use the term swim loosely, but... So we're about halfway up the slope. We are 30 nautical miles from Key Largo in this sand patch that we actually mapped last night. Considering there's no hard bottom here, there's quite a bit of benthic critters and a ton of squid that just keep passing by. Possibly migrating to mate. We saw this same behavior yesterday amongst these same squid up on the Miami Terrace. They were all heading north. Oh, what is that at 9 o'clock? It's flat on the bottom. I'm sorry, not 9 o'clock, noon. Now it's at 1. Oh, it might just be a tuft of sand. Never mind. Lame. I thought maybe we were going to get a big stingray. All right, we got a little bit of shrimp here at 6. Again, this bioturbation is pretty consistent through this area, which means there's lots of infauna or animals that live under the sediment. What do we have here? It's at right below laser. Possibly Glyphocrangon, a armored shrimp. Get a zoom on him. About 6 centimeters. Yeah, that's what it looks like to me. Got a little armored shrimp here. So it could be Glyphocrangon. That's one that's common out here. It's pretty cool. Yeah. You can see how tough it might be to get if you were to try to eat him. How hard it'd be to get through his shell. Yeah, he's about 6 centimeters wide. I'm good whenever you guys are. Whenever you're happy. Like this really soft sediment throughout this area. Likely a drift, which means the sand is coming over from shallower areas.
Ah, Roger. Sounds good. Thank you very much.
Oh, we have another small red shrimp there at 6, and some debris in these holes. There's an ophiuroid at 4, a little tiny, a large pink one. It's got 5 arms. Again, we have some of these active burrows ahead of us. This, some tall mounds and some divots. Yeah, it looks like some of these are, excuse me, old, inactive. There's an active mount or hole here. A couple active holes here at the bottom of the screen. 6 o'clock. I think I just saw one of those crabs crawl into one of them. Here's a chrysogorgid, likely at 4 o'clock. Yes, please. Yep, so I believe this is a type of Chrysogorgia we were talking about earlier, the species we've seen a few times in this dive. This is the one where Scott had made a comment about how they actually root under the sediment using a dripping type growth formation. It's not really roots, but it's a modified type of holdfast that keeps them stable in the sediments. It's an Acanella is the genus. And he's got a squat lobster, a pink squat lobster. And there again, we have this purple. Can you guys pan down at the very bottom of the screen? There's a purple blob there. I'm pretty sure it's a worm. And every Acanella has one of these associated with it at the base that we've seen today. I don't know. Yeah, I don't know what it's doing. I don't know why it's associated with it, but it seems to be that maybe they're territorial, or seems to be one per Gorgonian. This little polychaete worm, I think. Yeah, you can see his cirri, the hairs that stick out the side, they're quite shiny. And his head is facing the 9 o'clock. That's the front end. He's clear with the purple internal organs. A scale worm. And so that's a pollinoid polychaete. Thank you, Scott. And they are common associates of many invertebrates. It looks like he's kind of cleaning himself or doing something, moving his legs around. Thank you. Ah, it looks like a plate, yeah. Let's see. Scott says, best guess on the name of the sea pen is Calibel emnom, or Cofabel emnom. All right, noted. Scott, if we were to poke it and it was to draw itself in, would that help with your ID? This looks like an inactive burrow here. It seems to have filled in. There might be a couple spots of active burrowing on the edge. If there is a sea pen in the back, we can take a look at it. If it is a sea pen, are we capable of taking our arm out and giving it a little poke to see if it goes into its tube, into the ground? Is that allowed? Okay. Looks like a blob of squid ink floating by. Yeah. It may not. Not all of them are capable of that. So I certainly don't want to hurt it or damage it in any way.
All right, what are we looking at here?
Sea pen.
Nice.
Yep. Scott thinks Kelly Bellemnom or Kofa Bellemnom as the genus. He was requesting that maybe we give it a little tap to see if it would yank itself into the sediment, retract into the sediment.
The entire stock?
Yeah, so some of these sea pens will withdraw.
Ooh, let's poke it.
So yes, it's also possible it's Funcunella. Ooh, I'm going to mess that up. Funiculina. Funiculina. We need to see details on stalk before individual polyps. Yes, we're gonna give it a little tap here. It's very tiny. Let's see what happens.
Yeah, compared to the claw.
Just gently.
Oh,
there
it
it
goes. Oh, how cool. I had never seen that before. What a defense mechanism.
That's pretty neat.
So cool.
Thank you, pilots.
So does this confirm what we were thinking?
And he was wondering if only some of the genera are capable of it. That this one apparently is. Yep. So he's got a little top sticking out still.
Oh, that would be so cool.
Let's see if we can hang out here. Maybe he'll come back up if we don't freak it out too much. Yeah, if you got to move, go for it. So that's the cool part about being able to video this stuff underwater, is we can see their behavior, as opposed to when we get them in nets and whatnot and troll them off the bottom and do research that way, because you don't see behavioral behavior.
Yeah, I mean, do we know what kind of mechanism drives that?
I don't.
Or
Scott.
Oh, yeah, Scott's just asking now. How the heck do they do this? Is the question. So I guess we don't really know.
He says, I think surely they must have a preformed burrow under the peduncle, and essentially they ...
Just drive themselves underneath. They have to, they have to have some sensory thing for touch.
Squeeze water out of the peduncle, making it shrink.
I don't know. Yeah. I would be very curious as to kind of dig underneath it now.
So they sink.
See what it goes.
Quickly. Yeah, that's too cool.
Yeah, this one was pretty fast. It happened really fast. If you blinked, you missed it.
Yeah.
Luckily, we have our videographers that can rewind for us.
That's right. Instant replay on the screen next to me.
Beware, guys, now. We're going to have to touch every single one of them to see retract because, there could be slow versus fast, and what kind of scientists we would be if we didn't make sure. It's starting to raise, though. It has that third layer of polyps that we didn't able to see before.
Oh, that poor shrimp. He just took a nosedive. This is also why the pilots hate to collect sea pens. Oh, yeah? Yeah, and our lights, you can see from Sirius's camera, there's lots of squid. And so he's going to slowly come back up, maybe. If he doesn't move too much, we can head off.
Maybe the sediments, are deterring it also from coming back out. And Scott, I think those are individual polyps coming off. We saw this sea pen quite a few times before. I'm thinking it might have been some form of coral, I guess. Sea pens are octocorals. But we weren't sure. It did have individual polyps coming out, and then those polyps had their own tentacles, which then also had pinnules on them. So that's why I thought maybe it was closer to a coral than it was a sea pen, but not sure.
We'll get a better look at the next one without touching it.
No, we need to poke it.
See if we can.
I want to poke it.
We've got something at at noon. Oh, it's a tripod fish maybe coming in at 3. Yep, just right at laser there. Oops, he jumped away.
Yeah. The squids are now a nuisance.
Mm-hmm.
Ooh, is that another tripod fish?
I think so.
And I think we're looking again at Bathipterois. Bathipterois?
Okay. Can you post that to the chat room
Yeah.
so I can copy it?
Oh, are you doing the sea tube?
Yeah, I'm C-tube and taking notes for the end of dive. Yeah, he's cool.
Sea
So-
tubing.
Yep. This is a tripod fish. We saw this earlier. I love these modified pectoral fins. I'm not even sure if that's what they are. That shoot out front. Look, it's almost like he's got these like alien antennas.
Yeah.
He's like sensing in front of him. They're split, bifurcate at the end. How cool is he?
Yep, but we're seeing the typical characteristic that gives the tripod fish its name. Its pectoral fins underneath it are touching, and then the tip of its tail, of its fin on the tail.
Yeah, and some of the deep sea ones can almost sit like a foot off the bottom.
Yeah.
They're amazing. I don't know how well they can see. I would imagine not very well. Look how small his eye is. And really, what's the point of seeing too much down here? It's usually, there's no light except for us and bioluminescence.
Probably why those sensory tentacle organ thingies
Yep.
are
So
spread
big.
so far out,
Mhm.
so he can
like
sense things in the water column.
Like a satellite dish.
Squid interrupting.
Mm-hmm.
Ah,
Yep.
he's on high alert. And Scott is saying in the chat room that the sea pens that we are looking at, so that, as I said before, that they are actually they are related to corals. They are corals. They're in the order of Octocoralia. They're simply octocorals specialized for living in soft sediments. And then there's a different kind of sea pen called a rock pen that are specialized sea pens who go on back kind of the other way and look again for those hard substrates to attach themselves to. So it's not just the sponges and corals, the standard corals that we normally see on hard substrates. These are also sea pens that also go back, and those are referred to as rock pens. So thank you, Scott, for that information. Is that a different tripod fish, or is that the same one that keeps being scared off?
I think it's the same species, just a different individual. Because this one looks a lot bigger than the last one we saw.
That's true.
And I think there's another sea robin there at noon.
Yep. Oh, and then maybe another fish in the
cucumber?
back. We did see a cucumber earlier. I'm wondering if it's the same
At 9 o'clock.
ish.
So, yeah, we got a squishy cucumber at 9 o'clock.
Oh, it does look similar to the one that we saw earlier. It should have spines on it if we're talking about the same kind of species. We did not have a proper identification on it. If you could maybe take care of that one.
Yeah, so he's pink with these, like, palpate skin.
So the other one that we were looking at was very white and translucent
Okay.
versus this one's more pinkish.
Mm-hmm.
And it seems to have a lot of those spinal
Mm-hmm.
tabs all along the back, also along the edges where it
Mm-hmm.
attaches to the seafloor.
Mm-hmm.
The other one did have longer antenna, so maybe this is a different species, or they're retracted. They
It
can
could
be retracted, no?
Yes, I think
Yeah.
so. It depends. I mean, they can do all kinds of stuff. If you disturb them enough, they'll expel their guts. So
Can we poke him?
we can, but I don't want to see him throw his guts up. See, we have another one of those scale worms that we were noticing, maybe possibly the same as the one that was with the octocoral.
Oh,
This
yeah.
is probably his back end. I don't see any feeding appendages. So their feeding appendages will be at one end, and you'll see the little tube feet come out and usually suck the detritus off the sediment, or particulate organic matter off the sediment. They're deposit feeders, so they get their food from basically eating sand. All right, and
Any
then
of the microbial stuff on sand?
yeah, and their body expels the sand and keeps all the bacteria and detritus and dissolved, particulate organic matter.
Digest, yeah.
Mhm. Yeah, I believe this is probably the front end. I do not see his feeding appendages, though. Too cool. I have no idea what he is, so I'm just going to call him Holothurian. The best we can do. Yes, this is his front end. I don't see his feeding appendages.
So the other side might be the front end also.
Yeah.
Not sure. This one seems more rounded off where the other ones seem to kind of curl in at the end.
Yeah.
So not sure if that makes any distinctive traits.
No, I'd have to flip him over and look for his mouth.
We can poke him.
We could poke him. We try not to poke too much stuff.
I understand.
He's probably really soft too. It's really sensitive skin.
Probably. Back at some of the labs that are at UM near where my office is there's a lab specifically dedicated to studying aplasia.
Mm-hmm.
And what's really fun about them is that if you bother them enough, they squirt a purple ink. And, when you're having a particularly hard day, writing a dissertation, and it's just not writing itself, you walk over to the aplasia lab and you bother a few, and it turns, all the tanks, unbeknownst to me at the time, were all attached. So I thought maybe I could just disturb one tank. And they were all connected, and all of a sudden, they turned all very bright purple.
Did you upset everyone at the lab?
This was after hours, so I don't really know what happened after the fact. I just got out. I left it there.
You're not allowed to run around torturing anemones.
Well, I mean, there is now a lock on the door. So take that information how you will.
What do we got? Yeah, a little lobster. That's probably the back end. So there's his front door. Yeah, with a squat lobster.
A hydroid in the front there?
The little guy probably.
Yeah.
Or worm feeding appendage of some kind. Yes, you can see little worm tubes sticking up.
I was looking at this.
Oh, yeah.
Oh, and a nice pteropod shell.
Does he have, like, he looks like he's got a rubber band on him, but it's probably just some old detritus
Yeah.
draped over his arm. Like a scarf.
Or maybe some plastic. I don't know. Could be.
Oops. Oh, there's another C pen right there. We've got lots of these divots up here, some burrows. They're probably active burrows.
Yep. Ooh, what is that little blob thing at about now?
9.
8 o'clock, yeah.
Maybe
Could it be another octopus hatchling? Bob squid hatchling? Oh, please, that would be adorable.
Yes, I think so. A little bobtail, maybe? He's pretty dark.
Yeah. And chunky. Can we get the lasers on? Alright, we'll get a look afterwards. Seems to be trying to be active, but I'm not sure what's going on there.
They usually hang out in the bottom. I don't know what he's doing with his swimmers, but maybe he's threatening us.
Oh, we're scared.
I know. Terrified. He's so cute. Look how beautiful his
Intimidate
blue
us
skin
with
is.
cuteness.
I wonder if he's bioluminescent. If Mike Vecchione chimes in, we'll chat about it. Too cool.
Very nice.
Looks, he's just like thin layer of clear,
Yeah.
and then underneath that
Tissue.
it's silver.
Which I'm guessing can also change color as they please.
I would assume. I do not know what kind he is.
Chris is saying in the chat room, why are the fins moving so much? I don't know. Maybe it's trying to get up off the ground, trying to maybe get an act for swimming. It could be a hatchling. Mike Vecchione said that the size, I mean, we have to zoom out for the laser size, but it looks only a few centimeters big, which could be the average size of a hatchling. So maybe it's just practicing before, maybe it's getting its rhythm down before it officially starts to try to swim.
I don't know what it's doing.
I don't know. I'd like to see the front end.
Yeah, he's terrified. Okay.
Well, slightly bigger than the last one we saw.
About 3 centimeters, maybe.
Yeah.
4. Okay.
Yeah. Probably around 3 centimeters or so. You're expending quite a bit of energy, little guy.
I know, just sitting there.
Waving the fins. Scott Francis saying that it has gills on the mantle cavity, so perhaps it's ensuring that the water is mixed around so it's not kind of inhaling again the same expelled, I guess, carbon dioxide or oxygen around it, so it could be mixing that up.
Circulating its
Not
water.
Seeing the tentacles. Where are the tentacles?
They're probably tucked underneath. Yeah. He's like tucked underneath like this. You can kind of see it looks like his tentacles are tucked underneath.
Maybe he's trying to go into a burrow, or maybe something grasped onto his tentacle is trying to pull him in. Maybe he's trying to escape.
know.
I don't know.
I don't know what he's doing. He is super cute. He has really cool colors. It's like purple, shiny purple. Some maroons. These giant eyes. It looks like a squeaker toy.
Yeah. I'm wondering maybe if he is actively trying to escape something that grabbed one of his tentacles. I don't know. Doesn't seem like too much of a struggle.
He did just open the tentacles up a little bit. I hope it's not us making him swim around so much.
I think he was doing that before we shined the lights on him. Didn't look like he was drastically changing any behaviors. Okay. Well, I think we're good if you guys are. Really cool find. I'm loving the colors. Some more brittle stars along the seafloor. And we're still coming up quite a few of those volcano mounds, and then some other ones around it, some of these burrow hole entrances that don't always look like they have a volcano mound. So, we just passed over a sea star there, one of the cookie goniasters.
Goniaster. Goniasteridite. There's another one right there. If you could swing 3 o'clock to the right. There's a different kind.
Of sea star?
Keep going. Keep swinging. There it is.
Oh, yeah.
This one has longer, less connected legs.
Yep.
Looks like we have another pycnogonid in the water column. We can barely see it.
How does it look? Oh, no, it's not similar to this. I almost was having a...
I think it's a goniaster, though, so it's the same family of starfish we've been seeing before. It's just that the legs are less connected, so it looks less cookie cutter-ish.
Yep.
And more sea star-ish.
The central part of it also seems to be a very different color from the outer rim.
It's also raised, so we could actively be eating.
Yeah. I don't know if the sediment there is intentional to be somewhat camouflaged, but
yeah, or just drifted.
Quite a coating. Yeah.
I mean, a lot of sea stars bury themselves in the sand, so.
Maybe the current's taking care of it for them. When we pull out, can we get the lasers out? See a size average? Okay. Oh, it's pretty large, about 7 centimeters, maybe 6 centimeters.
We got a squid right next to us.
Oh, yeah. Scared into submission. Frozen in fear. It's a pretty large one, though.
Yeah, he is big. He's 30 centimeters.
Yeah.
He might be checking out our lasers. I don't know if they can
Or maybe
see
looking
that.
at the sea star.
It doesn't seem that the red lasers seem to bother the fish too much when we do these. The green ones will. The green lasers really have the fish just dart away from them.
Yep.
But the red lasers don't seem to bother. Hey, who's this? Who just chimed in?
Hey, this is Chris Ma.
Hey, Chris. What are you thinking on this
I was
guy?
just looking at your starfish with some interest. So the sedimented bottom and the habitat gives me hints, as well as the surface of the animal, that this is probably, it's one of two things, but I'm going to guess that it's a sea star called Pseudarcaster, which is, it used to actually belong to the Goniastridae, and I split it out with some genetic data a few years ago. But one of the reasons that I could tell was because the surface has these little kind of spiny platforms. They go by the name either tabula or papilla. They're essentially sort of like sort of table-like columns with spines or granules on the surface. And these are common in animals that bury themselves. In theory, what they're supposed to do is to kind of keep all of the papilla, all of the gills on the body surface clear, and catch essentially all of the sediment at the top. And this, in addition to several other adaptations, is thought to be part of a suite of things that makes this group of sea stars so successful in faunal habitats. They live, everything in this group, the Paxillosa, that lives in sort of sedimented, loosely consolidated sediment. So, they can be fairly nondescript, but there's actually very little known about them, and they're probably important in various ways, but seeing them alive is something that, again, we have collections of these, but we don't really see them in situ very much, if at all. So, so definitely, an important, everything you see with sea stars is important to me. So, think that as you will.
Can you spell his name for me, or post it to Twitter so I can grab it, because
sure,
I can't see it.
I'll do it on Twitter, but
okay.
It's basically Pseudarcaster.
Pseudarcaster.
Yeah.
Okay. Thank you.
And I'll try to throw it on there when I can.
Okay. Thank you.
Great. Thanks for the information.
Thanks for stopping. I'll talk to you later. Bye-bye.
Bye.
Bye. Seem right to be in the middle of a lot of squid here at the moment. Is that another anemone or crinoid we're passing over? Not sure. Yeah, just saw something as well. Oh, maybe a serpulid worm tube with some hydroids colonizing it. What's that brown thing?
I don't know.
Maybe some detrital material of some sort. Oh, with a little gastropod on it.
Right.
It looks like seagrass.
Oh, yeah, it could be. What is this bubble down here?
Pteropod shell.
Pteropod.
Very beautiful. land. If you're just tuning in here, we're off the southeastern United States, off Florida here. We're latitudinally very close to Key Largo, exploring some unexplored territory in this area, and it seems to be composed of a lot of this carbonate fine grain to coarse grain material, filled with a lot of skeletal material as well as some finer grain particles that we can't seem to make out. And we started at approximately 640 meters water depth, and we're working our way up slowly. We're about 610 meters now below sea level, about halfway through our transect here, going up slope, and you can see that in your feed 3, corresponding to where we are. We've collected a potentially undescribed species of a carnivorous sponge that's has not been recorded in this area before. So, and the Steph, the co-lead biologic scientist here, has also only seen it once before. So that is great hope in saying that potentially this is a really rare species that we don't see very often. We did see one other specimen here during our dives today. We saw some various fish and crustaceans, including a blind lobster and some various crabs. Fish, we saw tripod fish. We saw something that had stuck its head in the sand. Still not sure of eel-esque looking fins.
The finished Raja, the underworld skate, possibly.
Yeah, we saw quite a few shrimp.
Codlings.
Yep, and a sea cucumber, 2. Potentially of the same species, but slightly different color variations. We saw quite a few brittle stars, some sea pens.
Yep, Echinella. The only Gorgonian we've really seen is the Echinella.
Yep. We saw a few sea stars now. I think 2, maybe 3 in total. We have quite a few of these brittle stars that seem to be kind of splayed out and far larger than what we've seen on any other location or
We
dive.
have something at 9 if you guys want to zoom in.
It looks almost
It
like a
looks
bug.
dead center now.
Yeah.
Might just be a rock, but it'll be the first rock we've seen all day, so might be interesting.
Oh,
And
no.
it's just Thalassia. So this is just Tetraidal Thalassia, Tetraidal seagrass from the shallows. It's pretty common out here.
Yep.
And also really boring. So
We also saw munipsoid isopod. We saw 2 of those, and we learned that the differences between a sea spider and one of those isopods, the fact that isopods have an abdomen and sea spiders do not, and they're a completely different class of organism. And
both arthropods, though.
both arthropods, though.
Same phylum.
And it was definitely interesting to see how they work with their way through the water column and then potentially settle on the substrate sediments, potentially using the water just to have a transport mechanism using the currents.
I think I see a crab at 3.
We're just about to pass over that little guy there.
I think these are the ones making most of these small holes. I keep seeing them coming in and out of the little holes. I've seen only a few, though. There's a type of box crab. I'm not really sure what genus to put them in, or really what family to put them in, but they're probably similar to the same family as maybe the golden crabs we saw yesterday. That nice boxy body, but they have this really pinchy, like almost fine point chelate, which is their claw. See, so it's used for picking, that really fine-pointed picking claw. So they're not going to be they're not going to be like an attack predator. They're going to be more of a picking scavenger-like behavior, which makes sense down here, right? Oh yeah, what's that? We got something at 9 o'clock. It's squishy. Looks like it has tentacles.
Maybe an anemone.
Could be. Could also be a nudibranch. Yes, you can see the chelate hand, right? It doesn't have those sharp spines that a lot of them do, right? It's really more for picking.
Yep.
Like a pair of tweezers, where just the tips touch.
And it looks like it's quite hairy.
Yeah, he's fuzzy, huh?
Which might indicate, is that a worm that's trying to crawl off screen now?
I don't know.
It looked like it was crawling.
At 9, yeah.
Yeah, but those hairs look like maybe it was intentionally trying to trap maybe the sediments floating around it to cover better.
Could, because that would blend into the background really well if it was
Yeah.
covered in sand. They also, if they're burrowing, if they are the ones creating these burrows, you know, maybe it helps clean their burrow out, keep their burrow clean. This, yeah, clear thing, I don't know what this is.
Looks like an anemone.
Yeah. Could zoom in on him. Oh, yeah, it could be a tiny
Yeah.
holothurian. Little baby guy. Thus, I think I do see a tube going through him, which would be his intestines if it's a holothurian. Or a sea cucumber, deep sea cucumber.
I have seen a few structures in the sediments where it looked like the sediment had gone through a tube.
Mm-hmm.
So it could be these guys.
So that would be sea cucumber poo.
Yeah. But they're very small, so I
Mhm.
don't think it came from the larger one that we saw. So it might be of this one.
Mm-hmm.
Could also, the smaller pellets looking things could also be from shrimp.
Mm-hmm.
Shrimp
And also
pickle pellets.
if we saw sea urchins do the same thing. They create
Yep.
these really perfect round balls of poop.
No, pellets. No, pellets.
There's a shrimp at 3.
Now we, I think, passed over another...
Ooh, active burrow down here. Is somebody in that? Just detritus. That stupid grass. It's getting really old. Ha, that was a really bad pun. I do see feet. I see feet. Little pink feet. So, arthropod of some kind.
I don't know. It looks like some shrimp.
Mm-hmm.
Those are little shrimps that are head first in the burrow.
There's see the crab here.
Yeah,
this
the
white
crab.
crab we just saw. In this white one, but then we've got this I'm sorry, smaller hole. But then there's this deep pit that's got some other arthropod in it.
It looks almost like a mangrove seed that's been covered by
Oh,
some
yeah.
hydroids and stuff. And a little squat lobster looks like at the end.
Mm-hmm. A hermit crab. I think that's a hermit. Is that as tight as you guys can go? Okay. Oh, okay. So if you look in feed 2, is that being broadcast? No. Okay. So we can see it, but you guys can't. So we're looking at a down shot directly into the hole from a camera that's facing down on the top of D2, and they're going to zoom in on it. Oh yeah, it is a squat lobster, that little pink thing.
Yeah.
Yeah.
I'm not sure if that other thing looks like claws or if those are just two shrimps trying to...
I think it's claws like
Oh,
this.
the one coming from under the rock. Up on the top
Yeah, these
portion.
guys?
Yeah.
Yeah. I think it's
Claws.
It's claws, yeah, 2 chelate hands from the same critter.
Yeah.
Okay, we're good.
Thank you, pilots. That's a really cool feature. Technically, it's meant to see if there's something wrong, I guess, with D2 to get an interior look, maybe. But
They use it to collect
could
too
use this.
sometimes.
Yeah.
The down camera.
Oh, thanks.
Are the seas picking up? Or are we just facing a weird direction? It's getting okay. It's getting kind of rough.
A few bumps.
Mm-hmm.
A lot of these little spikes coming up are likely serpulid worms, either living or not. But they tend to form these like little casts that come up out, and then they crawl up the top and expose the little tentacles that they have, trying to catch some detrital material in the water column.
Use some of these anemones down here at 6. And those little holes. There goes a crab. Back in his hole. Oops, hi. Excuse us. Oop. Yeah, so if you don't know already, you can actually follow us on a lot of social media. We've got Facebook, Twitter, and Instagram that we post to. And you can also ask us questions if you like, and you don't have to ask us science questions. You can ask us questions about how a boat operates, or if you have questions for the pilots or the videographers, we can always get your question answered by someone on the ship if you'd like.
Yeah.
So feel free to reach out. We're on Facebook at Ocean Exploration Research and at Ocean Exploration Research-Education are the 2 Facebook pages. On Twitter, at Ocean Explorer, and use the hashtag Okeanos, and I follow that all day, so if you post something there with that hashtag, I'll see it. And on Instagram, at NOAA Ocean Exploration. It looks like we have something different here. What is that?
Is that a crinoid?
I think it might be. You can find me on my Twitter feed at Deep Sea Nerdy or at Deep Sea Nerd on Instagram and Facebook, and Kim is at Deep Sea Kim on Instagram.
Yep.
So it looks like Xenometra, the same one we saw, maybe, I'm guessing, because Chuck's not here, but it's similar to Xenometra.
So the one we saw on the Blake Plateau.
Yeah, but that wasn't on sediment. That was all on hard bottom, so that may be irrelevant, and it may be not. Who knows?
I'm not seeing the pinnules that go all the way down to the mouth.
All right.
So I'm not sure if maybe that was that new species that Chuck was mentioning, that he hasn't seen it in anything but purple.
All right.
You can see it's unstocked. It has a root-like feet structures that hold on to the sediments, and whenever they want to, they can just let go and swim. I'm using that in quotation marks through the water column. Usually they're using the current itself to actually transport themselves, but they can flap their arms about and kind of determine how high or low they are in the water column.
Right.
Nice little feature. Thanks, guys.
We have a question from Sackett Tribe on Twitter. What determines whether or not you decide to collect a species you find? Okay, so we are community driven, which means we have many scientists on land side who have a vested interest in seeing what we're seeing and getting research. So we don't collect anything for a specific project. So, like, you know, if a thesis student needs a sample of something, that's not for the community. We only collect for the community. So all of our samples end up at the Smithsonian in Washington.
Can we get a quick zoom in on that other feature that's sticking out right there?
If you're a scientist, you can actually check them out like a library book. So we target anything that could be a new species. We've got a, Chris.
Another one.
You might want to call in. This looks like a different one.
Really?
Yeah.
Looks the same.
That's a different species, I think. The bulbs are different. Sorry. Let me finish.
Well, to my untrained eye, yes.
So, can we hold here, guys? Let Chris get on the line real quick. Yeah, so if we find something that's a new species or something that is really abundant in the area, but we don't know what it is, or something we think could be an extension of a previously known species from a different area. So it would be a biological extension of their habitat.
Or an undescribed species.
Or undescribed, right. So those are the 4 criteria we try to follow. We also collect for the Aspire campaign. And that is, part of that is genetic connectivity. So there's specific species we're targeting for that, like Lophelia and Afrocalistes that we were targeting. So we try to pick up one or two of those a dive if we see them, and they get DNA sampled, and then they're that goes into the Aspire database
Yep.
to look for genetic connectivity among species. So those are basically our criteria. I think this guy might be new, and I think we might want to get him. But let me get Chris on the line before. Let's see. Oh, yeah, call me, Chris.
She just got back.
I think this might be a different one than we picked up before, but it's definitely a carnivorous sponge. I have never seen this kind. I don't think it's the same one, but I'll defer to the sponge ladies.
It looks like it has some hydroids growing on all over the stems there. Another polychaete hanging out on it as well. The bulbs here. Look maybe different than the one we said before, but again, my eyes are a little untrained. They look very similar. Almost look like little mushrooms.
Mm-hmm. So we're just waiting for feedback from Chris. I think she's probably trying to call in. Chris. What are you doing, lady? Oh, look at it. Look at that scale worm is moving. Hey, there you are. Hi.
Yes.
Is this different?
It's different.
Yes.
It's very different. If you see the size of the reproductive structures,
Can we get the lasers on real quick?
they are chromatophores. So there's the sperm is formed inside of this round structure, and then they are liberated into the water currents when the reproduction takes place.
Okay.
So the size and the arrangement is quite different.
Okay. So this is.
The previous that we saw, it's a different species.
We should collect
And
this.
the actual, yes.
Okay.
This
Yeah.
is really
Confirm collection, please.
a very important part of the in the terms for the sponges. You know, you guys are finding with the diversity of carnivorous sponges.
Yeah. Hang on, Chris, one sec. Do you think you guys could grab it with your claw and place it in one of the side buckets? Okay. You're gonna have to maybe use your Gorgonian snipper. Go all the way to the bottom. Yeah, Corocotter.
This one is slightly higher. You can see the previous one.
Yeah, he like sits higher up on the sediment. Right?
Yeah, different species.
Taller.
Probably con the lab.
001, 01. No, 02. This is our second one, right? We haven't done any more. Do you want to give it the same name as the previous? Or do you want me to call it something else?
SP2.
Chondrochalatica?
Yes, Chondrocladia.
Yeah. Okay. Thank you.
No, thank you.
So we're looking at maybe 5 centimeters. Yeah, you might, go underneath. Yeah, you might be able to get its whole, like, what amounts to be a root system. I don't know what's under there, but. Okay. Let's just do whatever you're comfortable doing. Let's just go that way. Keeping it as intact as possible.
Ooh, look at that.
Nice, thank you. Looks like a little Christmas tree
I know.
with snow on it. Merry Christmas, Chris.
Merry Christmas.
Do you need, so yesterday for the carnivorous sponges, we were taking a DNA sample and a small piece in formalin, and then the main sample was going in ethanol. Do you want the same thing with these guys? Is that the best decision? Small
Yes.
piece in formalin,
Yes.
small piece in DNA, and then the main.
As Nico, yes, one of those branchlets
Yeah.
that have a round structure. You can take one of those and place it in formalin.
You can
and then
see.
the rest can go in the alcohol. Really beautiful.
Are we seeing the little claws that catch stuff? Is that what I'm seeing? These little tubes?
I think those
okay.
are zoanthids.
Are they
on there.
zoanthids?
I don't think they're part of the main structure.
Oh, wow. This is either something different or part of a differentiated part of the sponge body. Yeah? So this reinforces that we have definitely a different species.
Yeah.
Awesome.
Wow.
And it looks like the tube structure, a lot of them retracted into that main solid stem structure because a lot of them were sticking further out before,
Okay.
more
So
lollipop.
they retracted a bit?
Yeah.
Yeah, it's incredible how complex can the morphology of the sponges evolve for reproductive and feeding and different feeding strategies. Really neat taking. Thank you so much. Congratulations.
Oh, well, Merry Christmas.
I'm sending it to Shirley in this moment to see if she can see them.
Okay. If not, we've got the footage. We'll shoot it over to her if she's busy.
As in you for
Oh, I think that's a jelly floating by as well.
Oh, nice. Ctenophore, maybe? So we're looking at starboard, outer.
Beautiful job, guys. Thank you.
Right? Wow, you got lots of, yeah.
You make it sound like it's a bad thing. I don't know.
Oh, the dirt? The geologist is intrigued.
That was great. We might have a whole new species.
Thank you so much. All right, so we're moving on. We're like a little over halfway up the slope.
Yeah, just about. We're not seeing a huge slope gradient based from D2 here. So it's really shallow as far as slope goes. Not shallow water.
Yeah.
We're about 610 meters below sea level. And you can get a great view from feed 2 of Sirius looking at it, and Sirius is about at 590, so sitting just about 15 to 20 meters above D2 to make sure it keeps an eye on it. And it also is that relay in between the ship and D2 in case there's a ship turn or something, that it acts as that kind of tethering balance between it, so we don't have D2 kind of jolt around or anything if something were to happen with the ship. So Sirius, not just for visualization purposes, but is very important as that relay in between, that buffer between the ship and D2. So Sirius does not have any sampling capabilities or anything. It simply has the camera. It does have some CTD properties. It can measure the water column as far as salinity, temperature, and oxygen levels. And we have D2 also has those capabilities built in, along with what you can see, the collection trays and the vacuum collection samples also, where we now, as a newer feature, is these canisters where you can actually suck up and vacuum in samples, whether they be of organisms or sediments, and then we can definitely take a look up them at top, and then they have, it's a fantastic mechanism that was only recently, earlier this year, added on. And D2 is approximately 9 feet tall and out of water weighs 10,000 pounds. So this is a massive piece of machinery here where it takes a lot of power, of manpower to really operate this machine. So our ROV pilots are constantly making sure that they're in communication with the bridge, where the ship is going, how we can relay this information, where the ROV is going, making sure all the mechanics are working just fine, nothing is overheating, and this is also why we cannot use D2 shallower than 250 meters in water depth, because the water is simply too warm.
Can I snap zoom that?
Oh yeah, right at dead center.
Okay. It's another
Sea pen.
I think it's another.
Oh yeah, carnivore sponge.
That looks like the first one we saw.
Oh, now I see the differences.
Yeah, they're quite different, even though they're
yeah.
just like these little, like, cylinders on sticks.
Yeah.
They're just morphologically quite different. So
Now I see it. Maybe when we get the samples up on board, we can really make out some differences there.
Yeah. Yeah, but you can see when we picked him up how they kind of stress out in the
Yeah,
little
retract.
yeah, they retract a bit. I wonder if we can get them up on the
In that tank.
put them in the video
Yeah.
tank and see if they inflate a bit.
Yeah, and what is that next to it? Is
That's
that
an
sea
anemone.
anemone?
I wonder if we can get the measurements, the lasers.
You want the lasers on it?
Yeah, it's
Yeah.
just
Yeah.
about to be there.
Yeah, it's about 5 centimeters wide, maybe 10 tall.
Okay.
Yes.
What about close up? If we see those filamentous projections?
I'm having trouble hearing you, Chris. What'd you say?
No, what if we can get a good close up
Yeah.
to see if it has the extra filaments that we saw in the previous?
Yeah.
We collect.
Yeah, so if we can get a good close up. I think we're pretty far away from it, so yeah, we're sneaking up on it now, so let's So I think this is the same species we collected first.
Yep.
And this is the third one we've seen. Versus the one we just collected, which I think is a different species. But it's the similar, these cylinders on the end of sticks. This alien antenna shape. Look at these. See, it's got these thin ones there with no cylinder on the end.
No bulbs.
Yep. Look, yeah, it looks like little light bulbs, doesn't it? Oh my god, what a cool sculpture that would be. If any artists are out there who know how to make how to make the cool lights.
Carnivorous sponges?
Yeah, carnivorous sponge
that
lamp.
light up?
That's what I want for
mean,
Christmas.
we can patent that. And if we could also then get a shot of that base, that organism there, Scott might think it could be a flabellate coral rather than an anemone.
Oh, okay.
And it does look like it has some hydroids or something. Also
Yeah, it's the
colonizing
same.
inside.
It's the same like small things we were seeing on the other one. So yeah, it might be a coral over here.
And a little brittle star also at the base.
Yeah, it's some kind of isopod possibly right there. Right at almost 6 o'clock.
Oh yeah, one of those worm isopods. Sorry, flabellum cup coral.
You happy, Chris? You get a good look?
Happy. Yes, happy.
Okay.
They'll have a long filaments and the same, very similar to before, the small filaments at the base of the stalk. Thank you so much.
You're welcome.
No problem. Thank you, pilots. Just goes to show you'll never know what you're going to see down here. Just because it looks like a lot of sediments and mud doesn't mean that there is an associated macrofauna here. Potentially 2 different species of carnivorous sponges. One that has been previously potentially undescribed and never seen in this area, and a second one that is potentially another species of that, of the Chondrocladia family or genus. Oh, looks like that shrimp got scared from the squid and then ran into a crab there. Can we get a quick snap zoom on that crab? It looks like at the base of that little depression there. We can get a really good view from this area of those little kind of volcano things popping out in various sections that are seemingly random are actually either entrances or exits of nearby tunnels that are being burrowed by associated crustaceans or maybe some fish or whatnot, and it's an effective bioturbation. So we call it bioturbation because the biota, the biology of it, is actually interfering and with the overall substrate in the area and impacting it. So turbation means the movement or turnover of certain elements and substrates, and this is now so biologically turbated, biologically disturbed in that area. Oh yeah. I'm not sure if that's one very large arm of the lobster and he only seems to have one. It looks like it. Looks like he's missing his other arm. Similar to what we saw before. It looked like when the shrimp knocked into him, he went flat as a pancake. Maybe as a defense mechanism, trying to lay as close to the floor as possible so he can't be picked up as easily. Lost one of his claws. It seems like, again, he has that hitchhiker on his back. What looks more like a shrimp now. Can we get maybe a tighter zoom on that? The little hitchhiker, the red guy. I think it is a shrimp. Not sure as to what kind. But we've seen now 3, I think, different squat lobsters with this kind of a hitchhiker on the back. Now, while a lot of squat lobsters are associated with particular biota, meaning cold water coral communities or sponges in the area, they like to kind of hold on and grasp and reach further out into water column to potentially grab more food. There are some species that are perfectly content on the seafloor, on certain either coral rubbles or sediments. It seems to be like this one is perfectly content on the overall sediment, and it seems that he has a little buddy hanging out with him. Of the different squat lobsters that we've seen in this area, this one seems to always have that same kind of shrimp hitchhiker. It always looks to be the same color and texture. I have not gotten a proper identification of that shrimp. So if anyone does know or has a good background in kind of the squat lobster shrimp symbiotic association, please chime in and let us know. This arm is incredibly long as far as the squat lobsters go. Yeah. Or maybe it's grabbing onto the other claw? I'm not sure. Looks attached. On 2 joints. Wow. That looks more like an abnormality than being normal. The claw looks twice the size of the lobster itself. I think we're good. Thank you. The claw looks to be almost about 30 centimeters itself. When we zoom out, we can get the lasers back on so we can get an estimate of the size of the claw there. Oh, another shrimp there with its swimmerets out. Really interesting. I have not seen a claw that large. And also for the average size, the thickness of that limb per the size of the actual lobster looks very unoriented. Can we get the lasers back on? So that claw in itself looks almost 20 centimeters long. Whereas the lobsters, maybe 3 centimeters, 4 centimeters.
That's right, turns. We can get a better measurement. Keep going.
Yeah. And the claw we were just talking about looks almost like it has an extra joint in it.
Oh, neat.
And it doesn't seem to have a second one.
These squid, I swear. He like purposely came over to spray sand on it. Like, have fun filming now.
Our videographers are probably getting annoyed.
I see a crustacean of some kind at 3 o'clock. Maybe another one of those squat lobsters. There's also an armored sea robin at dead center. We've seen already. You can see that a difference the presence of hard bottom makes as far as biodiversity and abundance.
Yeah.
Because here we have maybe the same 10, 15 species, and they're sparse, few and far between, and very few of them are actually attached to the bottom. Whereas in the hard bottom areas, we've got much higher biodiversity, which means more species types, as well as a higher number of individuals, so more biomass.
Yep.
And you can tell why they call this guy an armored sea robin. It very much looks like he's like an armored tank.
Yep.
That shovel nose, that's a really good view of him. We've only been really seeing him from the back before.
And his modified fins below him to look almost like crab legs. Lets him walk across the seafloor.
And then what I think are these barbels, he uses them as like a comb the bottom.
And he has that deep red color because, well, not because, we're not entirely sure, but it's likely that it's also a defense mechanism that red is usually the first to dissipate in the darkness. So it looks, if we were to turn the lights off, not only could we see almost nothing around us, but red would be the first one to kind of leave sight. So he looks virtually transparent in the deep ocean. So probably a defense mechanism that's there.
He's got some nice spines running down his back. And a collapsed caudal peduncle. He's got it squeezed. His tail. He's going to get up and move. You can see how he walks along the bottom with his modified fins. So they're a type of Scorpioniformes, which is related to scorpion fish. As well as sea robins. And you can see these modified barbels on his chin. The previous ones we've seen, they kind of use them like a comb as they walk, likely looking for food or sensing the area around them. Normally there would be no light, so he'd have to be doing all this in the dark. Thank you. They're quite common around here. We've seen maybe 10 or 20 on this dive. He's about 10 centimeters long, maybe a little longer. Thanks, guys. Now we're back on the sediment, looking at the bioturbation as we head upslope to the northwest. We are 30 miles off of Key Largo, in an area that was just mapped last night. We had no data for this area before. We were planning a slightly different area, but there was cables in the way, so we don't want to dive on the cables. So yeah, just because it's not safe for the ROV, so we try to avoid stuff like that. We are currently at 605 meters. Our max depth today was 640. So far this dive, this has basically been the same. So this bioturbated sediment with a scattering of some benthic biota. Ooh, we got a big hole coming up here.
Ooh, maybe we get something big in it. Ooh, that looks like quite the tunnel there.
Yeah, right. I wonder if this is a tilefish. That's very large.
Come out, come out.
Maybe we can get some lights in there and figure out who's living in it. So that's a good 20 centimeter hole. Maybe 10, 15 centimeters.
Yeah.
But this looks like the largest active burrow we've seen today.
Yep.
With a very distinct hole.
Entrance, yeah.
Yep.
And a very large depressionary feature, probably 1 meter wide.
Mm-hmm. So the only thing I've seen make holes like this that are this large is tilefish. Maybe a large Acanthocaris, which would be the blind lobster. But it's tightly packed, well cared for.
Yep.
So somebody actively lives here.
Maintaining it.
Mm-hmm. Maintains it.
I'm not seeing anything moving inside.
No, there's nobody in sight. He might be hiding from us. We are very bright.
Maybe going deeper into his network.
Mm-hmm. We might be loud. Are we loud underwater? How loud? How loud's the ROV?
The turbos or the turbines.
Mm-hmm. Yeah, if you guys know, are we loud? Pretty loud. Those squid are so rude. I just can't even with them anymore. I've had it. They're trolls. Like a perfect shot. Look and see if there's a back door. So that might be his back door right here.
Yeah, at the back end there.
Right there is probably the back door.
Just shy of dead center.
Mm-hmm. A pretty large hole right there. Yeah, it looks actively burrowed out, which would make me think maybe a golden tilefish or a Acanthocaris. Yeah, if there's a back door.
Know.
Yeah, that is pretty small.
Looks more like a depression. He could have an intricate network underneath the subsurface and have multiple smaller holes around to escape from or to go and tunnel through. So any one of these exits could be related to those. It can.
Oh yeah, there's another active burrow here. It's pretty large.
Yeah, it could be that back door. Yeah. And there's one also right next to it. Yeah.
Yeah, so these two could be connected too. It's really hard to tell. The sand's so
Could be
soft.
caved in.
Yeah.
Whatnot.
If we went to go dig it up, it would just collapse. So you have to fill it with like pour
Resin in it.
resin in it. Yeah. To get the shape of the hole and dig it up.
And we've done that part of some projects I'm not sure about all over, but in our lab, some of the professors have gone out and done such experiments to see just how intricate these systems can be. And I mean, you can have up to 50, 60 different exits. You're having tunnels within the tunnels branching out, this huge network system that can go up to 2-3 meters down into the soft sediment, depending how much sediment that package really is, how thick that package is. So, and it can go for meters and spread horizontally out throughout. So some of these tunnels might be connected and some might just be interwoven in between. We really have no idea of knowing.
I've seen that on social media, the video of them pouring like molten silver into anthills. They're pretty neat.
And it's a very similar trait, and it looks very similar to those that from anthills. So we have another one, maybe another Echinella. With a squat lobster. What is that? Not sure.
Yeah, another one of those anemones.
Snap-zoom. All right, squid. Looks like an anemone.
So it's an anemone.
Before it gets covered in some stirred up dust.
So that would be Edward Seed. Oh my goodness. Edward Easyday. It's a type of burrowing anemone.
There's so many of these depressions. Could be from that tilefish, could be from the lobster.
Yep. Right?
Yeah.
Yeah, like this little 10 centimeter hole to like this 2 meter wide divot thing. Depression.
So there's another question from Helen on Twitter. Does a sea pen already have a tube underground to hide in? So this was something that was discussed in the chat room when we actually saw the sea pen. We're not entirely sure.
Yeah.
I think is the appropriate answer. It could be muscular, where all of a sudden the tube kind of just sucks in on itself and like wraps around underneath the sediment. Or maybe it has another tube that it kind of penetrates further down into the sediment, and then when the pen retracts into it, it fills in then that tube that maybe goes down underneath. We just don't know. But Scott did suggest more of a challenge to see if possibly we might collect one of these sea pens. There's different genuses that are associated with the Gulf of Mexico, but they're not entirely studied yet. And so it would be useful to get an ID to determine if it would be new or not. but we would probably have to put the manipulator arm surrounding the sea pen and try to snap it closed as quickly as possible so that when it tries to withdraw, we catch it kind of midway. But could be, like he said, quite a challenge to achieve. So maybe we'll try it if we see one again. That's also up to the pilots. So we'll definitely keep an eye out. But to answer Helen's question, I mean, we really are not sure how this works. So if Scott has maybe a better answer, we can definitely chime in.
He said earlier, I surely think they must have a preformed burrow under the peduncle, and essentially they squeeze the water out of the peduncle, shrinking it, and therefore basically sinking.
Yeah.
But we really just aren't 100% sure on
No. I don't know.
Like unbury the whole thing, right?
Yeah.
Have you?
Interesting. We just passed by one of those squat lobsters that had a very long claw like we saw the other ones. I didn't see multiple joints, so I don't know if it was the same.
And Scott did just chime in again. He said, great question, Helen. I suspect, or is it Helene? Helene. I suspect the sea pen has already created a burrow or tunnel, and the peduncle is inflated to hold itself at the top of that burrow, and when disturbed, it can deflate the peduncle and fall into its pre-dug burrow. But there's no tube per se.
Yeah. Great question. Goes to show how much we still have to research and explore of the deep sea.
On top of the fact that, I mean, like if you're out here trawling, right, the way they used to do research in the deep sea before we had submergence vehicles, and where our archive data comes from, and a lot of our knowledge about benthic organisms come from, is from trawls. So going, taking a net and trawling the bottom of the ocean and then pulling out whatever you see. So, you know, if you're not going to catch the sea pen, likely, because it's going to pop down before you can scrape across it and pick it up out of the ground. So the only way to get one would be to do box coring, which would be
Also,
a totally different thing. But yeah, you basically
drop a box down there and it releases a latch, kind of grabs whatever is surrounding it in the sediment and pulls it back up onto the boat.
Right. So this deep submergence vehicle work is fairly new. I mean, maybe since the 60s, right? Really, in 70s. So we don't have a lot of video footage of the habitat, I mean, the behaviors of a lot of these animals. So a lot of it's unknown, right? So a lot of the stuff we see and collect with D2 is totally new. Yeah, new footage.
First time we've ever seen it, and especially maybe it's not a new organism, but it's the first time we've ever seen their behavior. Definitely, like if you were tuned in yesterday, we saw one of the lobsters practically fishing. It had a piece of organic tissue within one of its claws, and it was waving it back and forth, almost like tempting certain other fish or organisms to come closer and check it out. So while holding the other claws open and potentially just going to quick grasp it. So
yeah, no, I'd never seen Acanthocaris do that before. I have no idea what this is. It is a box. Yep. Of
pinto beans. It's pinto beans.
Pinito beans. Dos
or
pinos.
It's chips. Or peanuts. Oh, it's peanuts. That looks like a peanut.
Pinito. Isn't that Spanish for peanut?
Pinito? Yeah, well, it's like a baby peanut.
Baby peanut. Okay.
I think it's actually from Cuba because the whole written description is in Spanish there.
Yeah.
Pinito. Maybe
funny.
a cereal. Peanut cereal. So close, but no cigar. You just have to keep looking. And this is another deep hole. This looks really interesting. The fact that it looks almost like it caved in, but the walls still look pretty much intact here.
Yep.
So Scott also chimed in that says that the burrow that potentially the sea pen is forming is lined maybe with a mucus to help stabilize the sediments in place, similar to what I was describing earlier with some of the shrimps and other crustaceans that form also the intricate network of burrowing under the subsurface, where they use a lot of that mucus and stuff and tap the sides of the wall in order to make sure that the sediments around it are stabilized. And these finer grain environments are perfect for that kind of cementation that occurs with the mucus and the fine grain sediments, because if you have coarser grain sediments, that means you need to develop a lot more of that mucus to hold the structure in place, whereas the finer grain sediments, once there's a thin layer of mucus coating around it, they tend to clump together and really stabilize. A dead stalk of something. Maybe a former spicules of a hylanema.
Could be. Could also be the tubes of a Oceanapia.
Haven't seen any of those here, though.
Me neither. They're usually more reef associated.
We are about 2/3 up from the transect that we have previously described. This transect has been kind of one that was designed on the fly. So last night we only just acquired this mapping data, and very early in the morning, part of our mappers and Mike White got up really early to decide what would look like a really great area to dive into. And if you look on the dive transect, it looks like we're slightly off the transect here, but that was actually happened more by design, because it would have taken extra time to try to get exactly to that waypoint there, and we wanted to make sure we had the long enough bottom time so we can really get a good grasp on what we're seeing in the area here. So we decided a few, maybe tens of meters to the south there wouldn't change all that much of the actual environment of the facies that we're looking at. So we're following the same plan as the transect, just slightly further down south. Ooh. Is that another carnivorous sponge there? Ooh, yeah, it looks like the first one that we saw.
Yep.
So they seem to be quite popular in this area.
This is a fourth, I think, of this species.
I think so. Nice, thank you. And it's about, a similar size as the last few ones that we saw as well. Oh, yeah. Is that a cuttlefish?
Oh, I don't know what that is. I doubt it. Cuttlefish are only in the Pacific, so it's got to be an octopus or squid. Unless if this is a cuttlefish in the Atlantic, we are collecting it. I don't care what it takes.
Oh, it is an octopus.
Look how cute. Yeah, you can see how palpate his skin is.
Very different from the species that we saw before. So we saw bob squid before. This looks like an octopus, different kind of species. Do we have a guess on what it is?
No clue. An octopus.
An octopus.
That's the best I can do.
It's fairly small in size.
Oh, we can see his tentacles moving, his arms.
Yeah, no problem. This is a good view. And then after the octopus, if you want to pan left into that possible sea pen before we zoom out. This is nice. He looks like, yeah, his back end is quite palpated, but it doesn't look like it's solidified. Like, it looks like it almost can retract.
Yeah, so they can usually change the texture of their skin.
Yeah.
The same way they can change their color. So if you watch some shallow water ones that go from like rocks to sand, they'll real quick change the texture of their skin to blend in better. They're really geniuses at camouflage. It's kind of difficult to really wrap your brain around the fact that this is related to a clam.
Yeah.
Hey, is this Mike Vecchione?
It is indeed.
Hi, how are you?
And I can give you a guess as to what that was.
Oh, please do.
That was a Bathypolypus verdii.
Bathypolypus verdii.
Yeah, it's a pretty typical bathyal octopod. We see them on muddy sediments quite frequently.
Mm-hmm.
We catch them in trawls pretty well too.
Nice.
Because we can trawl that kind of habitat.
Awesome. Thank you.
Do they grow a lot larger than this?
It's not a real big species. I don't know how big that one was. I'm talking on my cell phone, so I can't look at the video. And
Okay.
talk at the same time.
Can you zoom out real quick and let's get a
I think
laser?
it's a few centimeters large. It doesn't look very large.
Yeah, he's like 5 centimeters, 6 centimeters.
Yeah.
Yeah. So. Awesome.
They get to about a foot long total length. So
Okay, well, that we with legs, so from mantle to leg tip would be about a foot.
the
Okay.
standard measurement on all cephalopods is mantle length. But,
Oh, okay, mantle length.
if you
Okay.
stretched out the whole thing,
Yeah,
they get up to about a foot long.
awesome.
Nice.
Yeah, so he's pretty small compared to how big he can get. Awesome. Did you see the bobtail? Was that what it is? Bobtail squid we saw earlier? The second one?
I saw one a lot earlier, yeah.
Yeah, you called in on the first one, and then we saw a second one. That looked different. He was
no, I
like
didn't see
purple.
that one.
Okay, so I'm sure there's gonna be footage of it somewhere. We'll post it. It had purple skin. It was really pretty.
It would either be a Rossia or a semi-Rossia,
Okay. Awesome.
probably.
Yeah.
Yeah.
If somebody sends me a frame, I can take a stab at it.
Yeah. I'll see if I can grab one from
video
when
team.
when the photos get populated, I'll send it to you. Thank you.
Okay.
Thank you so much for calling
You're
in.
welcome.
Have a good day.
Thank you so much for zooming on the cephalopods.
Yeah, they're my favorite. So I'm kind of biased.
Okay.
Thanks.
Well, all right. We'll talk to you later.
Okay, bye.
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 I like how he can keep moving, but he's keeping all of his tentacles still curled in. He's being very wary.
You can see his chromatophores, his skin, the little brown dots.
Pigments that can change color.
And stretch a leg out. He's very slow crawling. Probably apprehensive about moving around too fast.
Maybe it senses some food.
You can see how he walks with his suckers.
Yep.
Kind of like poke
Hundreds
out.
of little feet.
That are also tongues. So that's how they taste.
There you go.
Aren't you happy you don't have taste sensors on your hands? Ah, we've got that sea pen too. This one looks different.
Yep. This looks very different from what we've seen.
It's much more,
The tissue is a lot thicker.
yeah, chunky.
It's shorter, but the polyps are massive compared
Yeah.
to the overall size. They're facing in only one direction.
Yeah. Yeah, that octopus trying to squish himself all the way down. It's too cute.
What is that other thing that's just about out of frame? Yeah, you scaled up. It was further down. Also, now it's dead center, about almost just shy of dead center. 6 o'clock, that little speck there. I don't know if that's living or not.
Just zoom in a bit and
I don't know if that's
I don't know what is a schmutz, I think.
schmutz,
Yeah.
maybe a piece of stick or something with zoanthids on it or hydroids on it. Not sure. Oh, that's okay. Oh, we scared the little octopus.
Did he dart off? I missed it. I wasn't looking.
Yeah, just the biometer.
Mm.
By an octopus.
Yeah.
So it's okay.
Yeah, there's a sea pen, I think.
Yeah, if we can get a snap zoom on that one. Looks to be even a different kind of species altogether, what we've seen. These polyps are even larger. And these face more laterally rather than in one direction. Very long tentacles on each polyp.
Oh, a common name for that octopus is the spoon arm octopus, according to the internets.
I wonder what kind of sea pen this is.
So that's a sea pen. It's different.
Scott says he might think that they are, Cophobelemnon for the last two sea pens.
Cophobelemnon.
Maybe some more Thalassia grass here to tridal material. I'm passing over another crab or lobster.
All right. Cophobelemnon logged.
Got a little more crabs here. Oh, darting off.
Oh, you have a little bit of a slope. Like 2 degrees, 3 degrees. Oh my gosh.
Looks to be about right.
Oh, maybe a shark?
A catfish, a cat shark?
Yeah. Cat shark, maybe
A marbled cat shark? Oh, nice. Seems to be relatively small, maybe just about 10 centimeters, if not 1 centimeter over.
Let's look up.
And just hanging out at the sea floor.
I like the patterning on his back. It's really pretty.
It is. Really nice looking. Has 2 dorsal fins and some fins underneath as well, probably to stabilize it on the seafloor. Smaller than the size of these squid. Ooh.
Ooh, ooh, ooh. It's a siphonophore coming into view. Looks like he's about to crash on the bottom. And yep, he's there still. It's hard to see. It's okay. These obnoxious squid. Yeah. These obnoxious squid are dead set determined to ruin all your footage. There he is. There's a siphonophore. This is a colonial animal, believe it or not. And they're pretty fast. So the front end is actually facing down. And that's the bell end. So if we get close enough, you actually see the bells moving. That's what it uses to swim. And then the back end is going to be its reproductive organs and its digestive organs. There you can see the bell moving around. See how fast they are? It's crazy. Yeah, right. They do have stinging cells. They're a type of ctenophore. And they are colonial, which means if you were to take one of those bells off and separate it from the main body, it would grow a new one, live independently.
I think it keeps running into the seafloor because,
Yeah.
again, that bright light thinks it's maybe closer to the surface than it should be.
Right, and its natural behavior is to escape the light, which is usually coming from the top.
Yeah.
So
So
it dives down.
solution is to go down deeper. Lo and behold, the
Right here.
seafloor is right there.
Yep. I think I just saw that same sea cucumber pass underneath us too. It might have been a dust ball though. There we go, back on the shark. He's quite small. I think this is this. Did we see him already? The same species? No.
I don't think we saw one on this dive, but maybe
Okay.
yesterday's dive. I think it's that marble cat shark
Okay.
that we saw.
I think yesterday, yeah.
Yeah.
All right, we're going to start taking bets before, see how long the squid let us look at this guy. I got 2 minutes. Oh, what's happening here?
Oh, yeah.
It looks like a star eating something. A sea pen?
Is a starfish eating a sea pen?
I don't know. Let's get in there and videotape it.
Or, no, it's a
It's a
dead-
sponge. Yeah, it's on top of the carnivorous sponge.
Oh, that's
too
so cool.
Okay,
And that's a taller sponge than we've seen in any of the other ones.
So this is I think this is similar to the second species we picked up. And it looks like he might be eating that. This is pretty cool footage.
Yeah, the sea star.
Between
feasting.
Chris Ma and Chris Diaz, I think.
Who will call in first?
Yeah, we're going to have the battle out here between the sponge and the echinoderms. You can see the tips of his arms are curled up.
This is the most artistic representation I have seen of a sea star and a sponge.
Mm-hmm.
Yeah.
Incredible.
Yeah, I don't know if he's actively eating. We'd have to get... It looks like he's got his tube feet stuck pretty nicely to this little...
Yeah.
Is that Chris Ma, too?
This is Chris Ma.
Hey, Chris Diaz is on the phone, too, so you guys are going to have to battle it out. The battle of the Chris's.
No.
No.
The collusion of the creed. Probably Chris can tell us more. I've never seen this ever.
Well, this is certainly a first, I'll have to admit. So this is an Henricia. Probably not the same species that we've been seeing, but that's not unusual because Henricia tends to be fairly diverse, but without a lot of distinguishing characteristics. So, I mean, this one might be known or it might not. What we do tend to know about Henricia is ecologically, a lot of them feed on sponges. I can safely say this is the first time I've ever seen Henricia or any sea star feeding on a carnivorous sponge.
Mm-hmm.
So,
Wow.
you know, E2 brute. So this one might even have some of these sea stars have a large hyaline tooth on the underside.
Wow.
And this is thought to facilitate feeding on sponges. Usually, they're usually feeding on glass sponges or demo sponges. So it's, you know, it's the function isn't ever exactly been sort of looked at, but it's thought it might be perhaps to scrape away the tissue or something like that. But this is what when we were in the Pacific, what Chris Kelly used to call a 2-for-1. I mean, it would be difficult to be fair, it would be difficult to quantify, but this is a novel discovery. So, I mean, if you wanted to collect the Cladorizid, that Henricia feeding on it would be a plus for me, and I would make every effort to figure out which one it was if it was described.
Oh, okay. We think it might not be described, so we could, in theory, collect this. We've already collected one of these Cladorhizids. Yeah. I believe it was similar to the second one.
If there are other priorities, then that's fine. We can move along because Henricia is not identifying. Henricia is not something I like to spend my afternoons doing.
Oh.
But it's a very interesting. It's a very interesting observation.
Yeah.
Sure, and it's certainly novel. So you know, I mean,
yeah,
if you can, great.
If
not, don't worry about it.
Yeah, if we collected it and you could look at the stomach contents maybe and see if it's actually eating this carnivorous sponge.
Well, I can't honestly conceive. I mean, I don't think it could be anything else. I don't know why.
Why else would be on there?
Ophiuroids climb up on these types of structures to suspension feed. Henricia doesn't suspension feed, as far as I know. So, you know, there's only one reason for stars to climb up onto you know, sort of these perches, and that is to feed on them.
Yeah.
And there's a long precedent of Henricia feeding on sponges. When we were in the Pacific, I think that large Rosellid sponge, one that was the size of a Volkswagen bus, that had multiple Henricia on it with its stomach extended. So, you know, I would stake dinner that this Henricia is probably just feeding on this Cladorhiziid.
And this might be the first instance of that we know of.
Yeah.
Yeah.
I would say so. I mean, it would be interesting to dissect the contents and see what was inside in terms of what aspect of the sponge it feeds on and so forth. There's always more to be learned from a collection.
Yeah.
But it's definitely a novel observation, and simply the video I would walk away happy. But like I said, if the sponge was something you had not already collected and you wished to collect it,
Yeah, had we not
I would.
Yeah, had we not collected it before, it probably would have been a major priority to collect this now. But I think the video footage and potential identification of what it could be from the other species, the other sample that we did collect, is definitely worth looking at.
Okay, then I endorse collection. if that's what you're saying.
Yeah, but it was interesting to see that some of the tubular.
And Chris,
Ooh,
Go ahead, Chris.
Yeah, go ahead, Chris.
let me put my 5 cents. I coincide. I mean, I haven't explored these deep regions. Just now is my first time, and but honestly, it seems to be a novel, very interesting relationship. I mean, we know that the Henricias feed on sponges, but to have this star climbing in the tip of this carnivorous sponge, I'd say we might have a different species of Henricia. We might definitely, I can see this image in invertebrate textbooks,
Oh, yeah.
Something really shocking. Yes, it's really shocking. I would not miss the opportunity as a person that works in zoology to collect both the sponge
and the Henricia. Do you think we should get it?
But honestly, it's we know that this is very valuable time. However, as surprising the distribution and abundance of these carnivorous sponges has been in the area, well, this association, it's really shocking. I mean, and how it's the whole architecture of it, to see this sea star doing kind of an equilibrium, while I think it fits. And we will help with the speakers that you'll find, Chris, inside of the sea star. And it will be important to correlate whatever is in the sea star with the speakers of the sponge, So I'm sorry, but I don't ask any more collections.
You won't ask for any more collections? You promise?
Another thing, I would say.
How long would it take for you guys to grab this? 2 minutes?
Yeah, I mean, we can collect it and see if it ends up being a different diversity or if it's more of one or either of the two species of the carnivorous sponges that we did collect.
And we'd be getting 2 samples for 2 community people.
Yeah.
So, well, not for them, but that there's an interest in both sea stars and the carnivorous sponge. So, I can't. Yeah, if you go down, can we get it? Are you guys positioned for it? Yes.
Yes.
Pick it up. Your best bet is to take the Gorgonian snippers, go out the bottom, and try and keep it intact with the sea star attached, even though he'll probably fall off between the bottom and the surface, I think.
Probably.
Could we get the last to see the size of the whole of the sea star?
You want to see the what?
The size of the sea star.
Yeah, can we turn on the lasers before we start? Real quick. Yeah, sea star is probably like 15 centimeters. And the Clatterized, it's probably about the same, height-wise. Yeah, that's fine. No, you can cut it halfway. We got the root last time, I think. Oh, okay. Yeah. It's up to you.
Whatever you think you can grab the best with.
What'd you do last time? We cut it last
Cut
time?
it.
Okay. Yeah, go ahead and try and grab it with your fingertips. I think the trick is to dig it up without shaving it. You know what I mean? Right, and you just shave it. Yeah, I don't want you to shave it. So, oh, it's pretty loose.
Well, I think it's just flexible.
Scott says, I think you should grab the top where the sea star is, otherwise you can easily fall off when you pick up the sponge. Yeah, I would try that. Yeah. Yeah. It's up to you. You drive. I'm going to stop
talking. I think just snipping it right where you are is a good thing, and then just trying to lop it all in the bucket. If he's feeding, he might not let go very easily. Although, pretty shameful to interrupt a good meal.
That's good. Go for it. If he falls off, we'll suck him up with a tube. Maybe you'll hang on tight. Aw, it's like a bouquet
of living organisms.
Like balancing a plate on a stick. Oh, I forgot to write down mine.
It's a 2-for-1.
So this is going to be 3B. Oh, good.
If the sponge gets loose and devours your other cladorhizids, I apologize.
It's in a different bucket.
It's happened before.
He's in a different bucket.
Glad
it's okay.
he's all a completely different side of the ROV then. Good call, pilots. Subconsciously, you knew that.
Port outer.
Port outer.
And thank you to the GFOE people. I always appreciate your efforts.
Yeah, thanks.
Not sure if you guys heard that, but a big thank you from Chris Maw for all GFOE people.
Thank you, team.
And Chris, too.
And thank Chris to give the perspective of how unique it was, too.
Oh, absolutely, and I'm glad we got some of that really good video footage of it close by, and maybe the video footage of it in situ and in the lab will become part of textbook normalcy. And this is part of the reason why...
It's entirely possible.
Yeah.
The timing of this will be good enough that I actually have a paper on the Okeanos feeding in the Atlantic, and it's in review right now, but it's going to come back probably within a few weeks, and there might be enough time to incorporate their observations from this expedition into that paper.
Oh, that'd be great.
Wow.
Press. So, yeah, so this might even be in press by the end of the year or early 2020.
Nice.
Nice.
Anyway,
well, we
thank
look
you very
forward
much. I'm always watching.
Thanks, Chris. And thank you also,
I'll
Chris.
see you folks
Yeah.
in a little while. Bye-bye.
Bye. This was a great example as to why we do this. A lot of this stuff has never been seen before, never been happening. These carnivorous sponges was one of the first time we came across them in this area, and let alone to find an echinoderm, a sea star, feeding on one of these carnivorous sponges. It's Chris put it greatly in the fact that, I mean, it was free for all out here. It doesn't matter what kind of carnivorous species you are, the bigger and the better. Predator will always get its meal in the end, and this is definitely one of the reasons why trawling could never have given us this kind of experience. We probably would have seen both of these species. However, we would have probably at that point seen the echinoderm knocked off the sponge, never would have known that it was partly eating it. And because we get to explore now using the ROVs in certain areas, we get to see everything in situ, and a lot of these behaviors that normally that we've never even seen or experienced before. And it's just been a phenomenal experience to be part of this and to work on it. And I mean, I'm excited to be here. So
Right, and we the samples of carnivorous sponges are very rare. So even if we did have Henricia with stomach contents, there's no guarantee anyone on the planet would know what was in that stomach contents.
yeah.
And speaking of, looks like we got some fighting squid.
Squid here.
Right, so just because we have maybe a sample or two of Henricia with stomach contents doesn't mean we can identify what's in it. So now we know likely that Henricia was eating a carnivorous sponge, which is new to science. So big find for today.
Absolutely.
Highlight of the day, I think, so far. Not too bad considering we're on sand.
Which is not a bad thing. We've seen a lot of biodiversity in this area. I mean, not necessarily a hot spot. It's pretty sparse and scattered about, but we've definitely seen a variety of sea pens and other corals, echinella, different varieties of crustaceans, squid, fish, what else? I don't know.
Oh, geez, I've got a whole list.
Gastropods.
These ilex squid are probably the most
Octopus.
abundant. The octopus. The bobtail squid, we saw 2 of them. Bathypolypus bairdii, which is the spoon-arm octopus, we saw 1 of them. Pleodicus robustus, which is the royal red shrimp. Glyphocrangon, which is the armored shrimp. Geryonidae, which I think was those chasing golden crabs, similar to the golden crabs. Possible snapping shrimp. Pelagic shrimp. Sea robins, codlings, underworld skates, marbled cat sharks. This long-armed brittle star I don't have a name for. Well, Xenometricolium neris, which is the crinoid, the 10-armed crinoid.
Which we saw too.
Yeah, this is also known as a sea lily. This holothurian that had that light pink skin.
Yep. And also the white one that was also seen earlier.
Squat lobsters. We saw Acanthocharis at the very beginning when we first dropped down. And then this pseudo-archaster, which is a sea star. And Henricia, which is also a sea star that we just collected. Henricia.
I'm seeing a lot of these burrowing features here, this bioturbated sediments, where we see this a lot in the shallow water areas where a lot of the shallow water shrimp bury down deep, and the fact that this also translates into the deep ocean with a lot of soft sediments around is very interesting. We can definitely make that a comparable experience. And it just looks overall like you can definitely see all the bioturbation that's occurring, like all those little bumps and little volcano features that you're seeing here are the leftover piles of sediments that have been dug out from these tunnels, likely due to several species of crustaceans, possibly some fish species as well.
So here we got 3 critters right in view.
Yep.
We have this long-armed
Brittle star, which
star.
moved quite quickly.
Yep. And
Actually.
then a long-armed squat lobster. And then in the back there's an armored sea robin.
I wonder if he has that hitchhiker as well. Can we get a quick zoom in? Yeah, thanks.
A little shrimp hitchhiker.
Yeah.
Associate.
And one of our data manager actually had a comment on what kind of shrimp usually is that hitchhiker that we've seen on some of these squat lobsters. It's a cleaner shrimp, where it's likely that symbiotic relationship where the shrimp gets to eat just by simply cleaning whatever or microorganisms has then encrusted or attached itself to the lobster. And it's a symbiotic relationship because the lobster then gets all the crud eaten off and the shrimp gets to eat. So both positive relationships there. So definitely one to think about.
So the question from Patricia on Twitter is, when collecting samples, is there ever concern that cross-contamination of unique species might occur? So we do the best we can to not cross-contaminate the DNA of two different specimens. Unfortunately, we are not able to get down there with a little bottle of bleach and clean the claw. So when we do the sampling or subsampling, we will actually take a piece that's not from the likely area that's touched. So we would take a piece from the extremity at the top.
Yep.
We cannot help the fact that a Sea Star has touched a piece of that sponge that's out of our control. We just do the best we can, change gloves in between, and then bleach and DI water our tools in between. And then don't let the specimen touch the photography table until we've taken the DNA off and secured that. So we do make efforts. It is possible to cross-contaminate DNA really easily like that.
And mind you, we're also on a boat, and we're not exactly in like a very clean lab or anything like. I mean, clean lab, it's clean, but there are certain aspects of what you can identify as a clean lab, an ultra clean lab and we do have. There's always a risk, and including also in the buckets and things like that. But the pilots take also very great concern in making sure that each specimen is located in different bio boxes or canisters, and we try really hard not to cross-contaminate, and they know exactly which boxes have been used. So it's also, they take great care in making sure if there's a specimen that we collected that's also very light, that's likely to float away, they usually will keep it on one side of the box and then won't open it anymore to make sure that we don't have potential cross-contamination where it floats into another side of the box or potentially floats away altogether. So there's a lot of things we try to do that we can do on the ship that we always keep in mind to make sure that there's not cross-contamination. But just like anywhere, there's always a possibility. So thanks for that question, Patricia. Really interesting, kind of a small tidbit on how we operate here on the ship and what we do. And we go and collect these samples from the ROV. We bring them up to our labs, and a little glimpse of how we do all these collections. So we take always DNA samples of everything that we put in, that we bring up on the boat, and photograph everything. We have special ID cards. There's a great program that is being developed, and there's always tweaks to consider that our data manager is really phenomenal working with, and we put as much information as possible about the specimens that we collect in size, weight, abundance. If we're collecting a lot of associates, how many ophiuroids are on each coral, or how many gastropods are we looking at, how many star feathers and crinoids, and how many arms there are, and the color, all these little tidbits that we record while on the ship, while we're doing all this information, to try to just gather the most information as possible, because we don't know who might check out these samples in the future, and they might not be able to get that same information once it's preserved. And sitting on a shelf maybe for a year, maybe 2, maybe 10 years.
We have 40-year-old samples.
Yeah, exactly. So it's something that we always have to keep in mind. So we just try to record as much information as we can as soon as we grab the sample, hopefully also while still alive, and record all of that data, because who knows who needs what in the future, potentially. So definitely something we keep an eye on.
Is that long-armed?
I don't know if he's
squat lobster?
Long-armed.
Is it different than the one before? It's not quite as long-armed as the other ones?
Yeah, but
it's very
his
hairy.
legs are stout, His front claws.
You can
He's
see
sifting
him feeding,
through, stirring up some of the sands.
He's getting his mandibles are going crazy, his mouth parts. Oh. See something?
Yeah, us.
Yeah. Yep, see it. See how he picks up food, gives it to his next chelate fingers, his feeding appendages, and then gives it to the next one. And
Sifting through.
Yep. Fascinating.
Eating any detrital material caught up in the sediments or growing on the sediments.
And he's going to clean his antenna.
Oh, how cool. And his little hairs help him do that.
He's going to clean the other one. Oh, he's too cute. I'm going to eat the stuff off of it.
Proper hygiene is important.
He's cleaned his legs.
They got good stuff on it,
Yeah,
too.
he's got good stuff on his legs.
Very spiny and hairy. And those front claws are very stout. They're very thick.
I think he's got caught wind of us. Doesn't want to move anymore. No footage for you.
Well, we got some already. I think we're good, video, whenever you are. Or pilot, rather. And we've slightly gone upslope here. I mean, it's not a very high degree slope that will be that we've been scaling over, but I do want to point out that there is a slight temperature difference. We started at 6.2 degrees Celsius at the base of this slope here, and it's now 6.5. So it's a 0.3 degree difference, which in a lot of contexts is not very much. However, it can mean the difference between certain biota living there or not, and usually if we see a drastic change in temperature, we have that shimmering effect that we saw quite often on the Blake Plateau there, where we know the temperature changed quite vigorously from not just a couple point degrees, but a couple degrees in its own. And that can really change the overall environments that we get to see down here in the deep ocean. Ooh, another Hylanema.
Yep.
Ooh, some zoanthids on it. You can see those spicules, those silicate spicules coming out from underneath the sponge.
Yeah, it looks like it. Zoanthids, yep. So that's his silicate stalks. There's spicules, which are normally teeny tiny ones in the cell. You usually have to have a microscope to see it, but this species grows really long ones. It's used as a stalk. So it looks like cotton candy on a stick. Looks like our squid cleared out. I mean, we're in a
Don't
lull.
jinx it.
Maybe we got too shallow. They're no longer here. And it disappeared
Maybe.
yesterday when we got as we were
little
getting
shallower.
shallower. And we're just a little too shallow for them anymore. Like literally exactly what you were saying. Like 5 seconds ago. Oh, the habitat can change. Animals can change.
Get to see so much of that depression stuff. What is that little creature hanging out there? Is that another frogfish?
I think it's another
Oh,
frogfish.
grumpy face fish. It looks like it.
He's a little 5 centimeter frogfish. Yep, he's another one of these angler type fish. They stand on the bottom and walk around. Usually have a lure between their eyes. I do not see his lure, though. He may not
Maybe
have
they
one.
haven't developed it.
Also, a lot of these, like batfish, like ones have it on their nose, on the tip of their nose. We used to have one at the aquarium when I volunteered, and you would take a little dead piece of shrimp and wave it in front of him, and he would take it as a little tiny lure and try and lure this dead shrimp. It was ridiculous. They're so cute. Yeah, so that's the batfish like.
Yeah, he has a lot of little pinnules on his underside. Kind of looking things. Not sure if those are spikes or not.
Yeah, they're barbly.
Yeah.
Camouflage barbels.
Okay. I think we're good. His eyes are quite blue.
And some of his skin's pretty clear. It's translucent a little bit.
Oh, you jinxed it. We have the squid back.
Oh, man.
What is that dark object ahead? If that's another burrow, or is that an organism?
Baby
Or is it trash?
Yeah. Schmutz? Is it ink? Squid ink. Yeah, that's squid ink.
Squid ink.
Mucusy squid ink.
I'm very intrigued by these large depressions here. What kind of organism could have done that? I mean, it could have been that blind lobster or the tilefish, but I don't know. The blind lobsters almost don't look large enough to really create these.
And I haven't seen any tilefish, but they're usually really shy. So they're probably terrified of the ROV. It's very difficult to find them usually. But this looks like a typical like tilefish looking hole, right? It's usually a front and a back door. That could be the back door. It's pretty long though.
Potentially. I don't know. And for those of you just tuning in, we are at the Key Largo deep area here. We're just south of the Miami Terrace, diving this previously unexplored region here. I'm Kimberly Galvez from the University of Miami Rosenstiel School of Marine and Atmospheric Sciences. Oh, I think there's a little sea star.
I'm Stephanie Farrington from Harbor Branch Oceanographic Institute at Florida Atlantic University.
And we are your science leads for this portion of the expedition for this month. We had a few hiccups earlier in the week due to some weather and unforeseen circumstances, but we're back diving now as of yesterday, and we escaped the weather out on the Blake Plateau, and we are here south in the Straits of Florida.
We've got a Goniaster, or Goniasterity type sea star. I'm not sure of the genus. I'm sure Chris is watching. He'll chime in and let us know.
And he's about 5 centimeters wide, if I got that right on the lasers.
It's one of those cookie stars or ravioli stars.
Just about maybe 4 or 5 centimeters wide. It's an interesting find. So it looks like potentially what could be a barren area is actually quite filled with a lot of biota here. I mean, it's not necessarily a hotspot of diversity, but we are seeing a variety of species and different organisms that thrive in this area specifically. Ooh, what's that little thing? Right at the 6 o'clock there.
Another bat? Another batfish or frogfish? Oh, it's a tiny batfish or frogfish.
Oh, another one. Oh, they're
so
grumpy
cute.
faces.
They're so cute.
He's about 10 centimeters when including the tail. We're good, thank you. And it takes a whole lot of manpower to make these explorations happen. We have 49 crew members on board. Ooh, I think we have another isopod. Munopsoid isopod. Now these are not sea spiders.
No, but they're very tricky. They look like sea spiders to me.
Yes.
So.
Which is for the untrained eye. Very easy to confuse. We've
See through
learned
the snow. Oh, yeah. Okay.
just kind of gliding along with the current there. You can see kind of how gentle this current system is in this area. It's not being whisked away by any sort of whirlwind of current systems there.
All the sand we've kicked up.
Ooh, might be going down to settle. And there are several characteristics of why it's also not a sea spider. First, that it has 6 legs. Second, that it has an abdomen. And we normally don't see pelagic sea spiders, if I'm not mistaken.
I don't usually see them, but I don't know how common they are.
Yeah. And these guys are not necessarily living their entire lives in the water column. It's likely that Mike Vecchio said earlier today that they are likely just kind of using the water column and the current system to transport them to another location where then they can keep going on or searching for food or whatever their purpose for moving on is. Possibly to also escape from a predator. I don't know. Oh, got some more ink there. And like I was saying before, this ship is manned by 49 people on board, crew members from everyone that navigate up on the bridge to the ROV pilots that you are manning this ROV that you are getting great footage from here, to also keeping the ship running. We have a lot of ship engineers as well as those that have to also feed everyone on board in the mess hall. And these are quite talented people and also videographers that are here that give you also some great footage that you can go ahead and look at all of the highlight videos and what you see on the NOAA website. And so a big shout out and thank you to everyone that can make this entire exploration possible. We are also, the Okeanos Explorer is the only federally funded ship specifically designed for ocean exploration, for deep sea ocean exploration. So that is a huge achievement that we get to do all of this and use the advanced technology that we are capable of having on this ship and providing this public data. So any of this data that's being acquired, any of the video footage, the still images, the bathymetry is all available to the public online for whatever use they need. We're doing this for the general science community and maybe not just the science community if you have different needs. This information is all online. You can definitely use the NOAA Atlas. Oh, we have a crab right in the center of the screen here.
It's
Maybe that golden crab?
Yep, likely Chasion fenneri, the golden crab. Yep, that's my guess. Looks like he's got
Is he
something.
carrying something?
Yeah, looks like he's holding something. Maybe a female. Yep, there's a female he's got a hold of. And they are hanging out, traveling together.
I don't know if that's willingly.
Yeah. It's a passive aggressive.
Can we get the lasers?
Yep, so we have a mated pair.
Oh, quite large.
It's about 10 centimeter carapace. Maybe a little bigger.
Maybe a little bigger. So when they mate in the end, do we know who carries the eggs or if they plant them on the surface or they carry them with them?
I don't know if they, I think they probably do carry them. That's what, in their little pouches underneath, that's what most female crabs do, is hold them in their, abdomen area. But I don't know about the species specifically. I didn't study their breeding behavior. I just studied their distribution in this area. So the male is the larger one.
Kind of frozen in place, probably because of the ROV, the light shining on them. Thanks, pilots. We're ready to move on when you are. Another squat lobster behind him. Ooh, and I think there's another carnivorous sponge out about 9 o'clock there to the side, coming in dead center. Yep, right now in the center there. Oh, just a snap zoom.
There's another one, a couple of frogfish too.
Oh, yeah.
Yep,
Yeah,
that's the
that
same
seems
species.
Like the same species that we collected with the same species we collected with the Sea Star, and also that first one, I think. Thank you, pilot. Oh, yeah, what is that? Is that another sea cucumber? I think that's another sea cucumber. Holothorian.
So the only thing I can find on Chaseon reproduction and eggs is from a paper in the Northwest Atlantic deep sea red crab, Chaseon quicquidens, which is its relative population before and after the onset of harvesting by Whale, Wahle et al. And the only thing they mention is that whether female red crabs are able to fertilize more than one clutch of eggs between molts, as lobsters can, is unknown.
Oh. Oh, here's that Holothoria. Almost looks exactly the same as the other species we found, that slight pinkish color. Okay, Scott says in that, I'm not sure, did you see what Scott said on the chat room? Well, he said in the case of these crabs, the male will be waiting for the female to molt. When she does, he will fertilize her eggs, which then become the embryos, and she holds the embryos under her abdomen using the abdominal appendages. Okay. Oh, there's another Hylanema there.
Oh, I believe the South Carolina Aquarium is live streaming until 3:30.
Oh, cool.
Would you give a shout out to anyone watching at the South Carolina Aquarium? We can do that. Hello, South Carolina Aquarium. How are you today? We're really glad you could join us. We hope you're enjoying the dive.
We're here on the Key Largo Deep. We're south of where we were yesterday on the Miami Terrace. We've come across quite a bit of unconsolidated sediments here.
This is Hylanema again. This is that sponge that looks like a cotton candy on a stick. Again, we've got these isopods associated with it. We saw these isopods before.
Yep.
Associated with Hylanema earlier in the dive. Don't know what their name is, what they're called.
Yep.
The best I can give you is isopod or amphipod.
They have 4 antennae.
Mm-hmm. They look like little crowns.
Yeah.
Their head.
Yeah.
Amphipods. Scott says amphipods. This sponge is really unique in that it's a glass sponge, so a hexactinellid sponge, and its spicules, which are usually used to hold its tissue together, actually grow on this one all the way down into the sediment. So you can see the amphipods are actually standing on these long spicules that the sponge creates to hold itself up out of the sediment. And those spicules are so fine they can be used as fiber optics. They're silicious.
Yeah, and if anyone at the South Carolina Aquarium has any questions for us, feel free to chime in. We have various social media reports that you can ask your questions to. They don't always have to be science related. They can be something that you can also ask the ROV pilots or even the bridge or anyone else on our videographers or anyone else on the ship here. So we have 2 Facebook groups that you can ask questions to at Ocean Exploration Research. Also at Ocean Exploration Research-Education. You can follow the NOAA on Twitter at Ocean Explorer, and you can use the hashtag Okeanos in all posts, and we'll try to give you guys a shout out or answer your questions if you guys have any. There's also an Instagram you can follow at NOAA Ocean Exploration. And you can also use the Okeanos hashtag, and we'll definitely look those up as well. Steph and I also have personal social media aspects that you can also follow. I'm on Instagram at DeepSeaKim, and Steph is at, I don't know.
On Twitter.
On Twitter.
At DeepSeaNerdy. And on Instagram and Facebook at DeepSeaNerd. And use the hashtag Okeanos. I follow that all day on Twitter. So I'll probably see your questions. Do we have a fish person online today?
I don't know.
Okay. There's a question from the good doctor on Twitter. When I've caught shallow water sea robins closer to shore, they usually make a croaking noise similar to a little frog. Do the deep water species also make noise? Also curious about whether the frogfish make a noise. They kind of look like they should. I agree. They do look like they should. I don't know if they make noises. I have never picked them up out of the water. We usually don't collect fish just because they're difficult to A, collect, and B, some of them have swim bladders, and when you bring them up, they bloat.
Pressure.
Yeah,
the
so
pressure.
we're not big on collecting the fish. I can reach out to Andy David at NOAA Fisheries and find out for you. Let me shoot him an email. If he doesn't respond by today, I can definitely get back to you on Twitter when he does. So I will ask him for you if we don't have a fish person online today.
Yep. And that goes to show how important our onshore scientists are as well. We're not experts in everything that we're here. Steph and I personally am a marine geologist, and I focus on carbonate environments within this area specifically. And Steph is a deep sea biologist. And even though she covers a wide array of different types of organisms down here, your specialty recently has been focused a lot towards cold water corals and sponges. And
mesophotic stuff. I've been really doing lots of mesophotic. So shallower than this.
Yeah, slightly shallower than this. Mm-hmm. And she has personal hobbies for cephalopods, so she knows quite a bit of those. But we can't know everything. And with each new dive, we're potentially exploring and discovering new species of different organisms here. So we rely on onshore scientists and other experts to chime in either in the chat room or on call, like Chris Smaw has, if you have joined us in the past, to talk about even the sea stars that are here. So we gladly welcome anyone chiming in or relying on that kind of information through the chat room or anything, because I don't think anyone can be an expert in everything. And it just goes to show how important everyone is in contributing towards the overall exploration of these cruises.
There's no shame in not knowing everything. That just means there's more to learn.
Exactly. And Scott says those amphipods that we saw earlier, those pinkish ones with the ones that look like they have little crowns on them, are likely gammarid amphipods.
Gammarids.
From the family... Oh, I shouldn't have started that sentence.
Ephemeridae. If humidity. I shouldn't have started that. I regretted that as soon as I opened my mouth. So Gamarita would be the genus. All right.
And there we have another small little crab there. Ooh, what is that pinkish thing that we have at about 10 o'clock also ahead? Off to the port side. A quick snap zoom. Oh, it's moving.
That long-armed, yeah. One of the Galatheads, one of the squat lobsters.
Oh, yeah. Backing up into its tiny burrow there. Okay. Those with the more robust arm structures there, the one that we saw feeding earlier on the sediments. Thank you, pilot. And like I was saying earlier, it takes a lot of people to man and operate this entire vessel so we can do the research that we're doing here.
And months of planning.
Months and months of planning. And so we want to introduce our front row here of who we have on our ROV pilots. If you guys have the time to introduce yourselves really quick.
Sitting in the front row, operating the ROV and cameras. I am the pilot, Levi Unima. To my left.
Yeah, Mark Durbin at Navigator.
To my right.
Lars Murphy and co-pilot.
And in the back is Caitlin Bailey doing video.
And we are all with the Global Foundation for Ocean Exploration.
Great, thanks. And this is just one set of the pilots that's here. So they do shift changes because operating this quite intricate piece of machinery is a hard job. So they do take shifts to relieve each other from duty quite a few times to take a break so that we can keep on going here. Ooh, what is this that we have here? Is this some debris, some old fishing line or something that we have? Yeah, it looks like some old fishing line here.
Looks pretty clear above you. I think it might just be on the bottom.
Oh, another one of those carnivorous sponges. And a few shrimp. Yep. Really nice looking sponge. I'm very excited to pull these up when the ROV comes up.
Right?
Absolutely. We want to make sure we stay clear of the fishing line. Don't want to get any sort of entanglement there.
No, definitely not. We're hoping when we have this, well, our videographers have this fancy little tank they have to make it easier to film like pelagic things that are living or behavior of living organisms that come up. So I'm hoping we can put these carnivorous sponges in there and maybe they'll inflate and
Yep.
they can get really good footage, close-up footage.
I mean, and considering this is new species, that would be great data to have.
Oh, yeah.
I mean, what that we had in situ in the environment from the ROV, but really high quality photos from the lab would also be really a good advantage. What do we have here?
There's something pink. 9 o'clock.
It's another grumpy fish.
This looks like a tonguefish.
Oh, you might be right.
Maybe. Maybe a worm.
And a soda can. Oh, what is that?
It's
It doesn't
got-
look like a fish.
Mm-mm.
Is that a sea cucumber?
It might be sea cucumber. Looks like we got a natty ice or something over there.
It
or
looks
purple
like it
soda.
has
Crepe soda. Yeah, it looks like a sea cucumber. You can see
some
his
fenestra
tube-
on the side.
2 feet underneath.
Ah,
He's pink and squishy.
I want to poke him.
Right. Boop.
Copy that. now,
Scott
thank
says
you.
maybe a scale worm. I don't know. It seems quite large to be a scale worm. But do they get that big? I'm not.
Oh, they
familiar
get
with-
scale worms get.
Ooh.
Yeah,
Okay,
gross.
can we get the lasers in case?
But scale worms usually have like these really distinct like the big ones do have like, real distinct scales. Yeah.
Okay, so about 10 centimeters long, maybe. Oh,
He says it's a worm. 100%
yep,
certain
scale
it's a worm.
Okay,
We will take your word for it.
and we will not be sampling it
Nope. I am not touching it.
because now you also know what it is. So
I am not a fan of worms. It's so silly because like I like all invertebrates except worms and roaches.
oh, the isopods are just so cool. I'm okay with worms. I'm just not good with things falling apart in my hands.
Oh, yeah.
And one of the main questions that we do get in general for a lot of people that maybe aren't particular to the whole science realm of this is how do we end up in the positions that we in? So, Steph, how did you end up to where you are today?
I have a undergraduate degree in marine science and biology from the University of Tampa, and I have a graduate degree from Nova Southeastern University's Oceanographic Center. It's a master's in marine biology. I did my master's on the biogeography of the Straits of Florida. So basically I mapped all the critters that were ever found past 150 meters between Key West to north of the Bahamas, and did a correlational study on really a meta-analysis on why things lived where they lived. So that's how I got my master's. And I did that under Chuck Messing from Nova. And then I was on a ship in 2005 and 2007 called the Seabird Johnson with Harbor Branch. And I met John Reed.
Copy that. Thank you.
And Chuck said to me, Dr. Messing at the time, said to me, he introduced me to John Reed, and he said, this is John Reed, and he's an expert in deep sea corals. And I thought, oh, I'm going to get a job with this guy one day. So I graduated. I didn't have a job for a long time, so I had to live on my uncle's farm, where I shoveled manure for 7 months, and then found a job opening at Harbor Branch. And I applied, and I got the job at Harbor Branch. So I've been there for 9 years, I think. So I'm going into my 10th year at HBOI. So that's how I got here. And Kim ran out of the room, so I'd ask her. She's getting her PhD in geology from the University of Miami. Oh, we're going to fight. I think she went to go get chocolate. You abandoned me. I was trying to stretch that out before you came back. So
I didn't abandon.
we need our chocolate break. We run out and
yes.
eat chocolate every once in a while. Keep our blood
It's
sugar
3
up.
o'clock.
I know.
Have that little break.
Yep. It's either nap time or chocolate time.
We can't nap on you guys.
So
But-
yeah, that's how I got to be here. Go for it, Kim. How'd you get to be here?
It's not nearly as interesting as Stephanie's story.
Well, I didn't go that deep into it.
Well, she's been also doing, been in academia and in science a lot longer than I have. But I actually started off, I always loved the ocean. I grew up in Miami, so it was kind of a necessary thing to always go and experience the water, and my family was always keen on making sure that we got to experience the best out of life that there was. And how do you tire? Oh, that opioid did not have a good day.
No.
Oh
man,
he really got chewed up. I wonder by what.
Ooh.
I don't think he can regrow back from this point.
Right.
Something definitely snacked on this.
Maybe a squid got him. That's weird. Why is
Maybe.
is that branch? It's got to be two different legs.
Yeah, I think that's just another half-eaten leg.
Yeah.
Ooh, that's fun.
Yeah.
Eat or be eaten. The way of life. But yeah, so we went swimming quite a lot and snorkeling and kind of experienced the ocean quite a bit, and I fell in love with what that was. And you would think that's something I would jump straight into a career with, but I was a little hesitant to focus all of my STEM field of interest in the marine realm. So I got a general degree in biology for my undergrad at the University of Miami. And then I started to work in the geology lab during my undergrad to try to gain some more lab experience and to work with some graduate students to potentially get some good networking in and to not necessarily learn a whole new field, but gain that experience for the intention was to always move towards also a more advanced biology lab to work in.
That's another one of those worms.
Excuse me. And
Another
I think
scale worm.
another scale worm there. Looks just as pinky as the other one. And, but it ended up never working out that I went to the other lab and I stayed in the geology lab. I got comfortable and I got to learn a whole bunch of new techniques and new machinery and how to work different things. They had an abnormally large amount of trust in me straight off the back to work a lot of these machines, and I became so intrigued that I got to be so hands-on right from the start that I really stayed and ended up when it was time to graduate and potentially start applying for grad schools. I knew that this is somewhere that I could be happy going actually to school and not just working in this environment, working for the grad students, but being a grad student and being on that cutting edge of research and giving my 2 cents and figuring something out and contributing towards the science world in there. And during this time of when I was going back and forth about applying, I thought, okay, if I apply, I have to come up with some good reason as to why a biologist should be in a geology program. And I ended up figuring out this underwater realm of cold water coral systems. And corals have always overlapped with the geologic sides of things because they leave an excellent record and they can tell us through geologic time how certain evolutionary periods happen, as well as they can shape and form the seafloor on the shallow level as well as the deep floor, as a deep sea. And I wanted to use that kind of intricate systems and figure out what could actually happen and how I can integrate my biology with the geology program that's there. I did so much research. I reserved an hour of my boss's time. I went straight up there and I wanted to give him a whole monologue of how I would be great for this and how this kind of project would suit me perfectly. And as soon as I went up there, he was like, "Oh, fantastic, you're here. I have an idea of a project that you can work on as a graduate student." And of course, it was the same thing that I gave him was about to give him the whole speech for. So I was robbed of my speech. And I asked if he still wanted to hear it, and he said no. We already agreed, so there's no point. And so then I started in graduate school, and I started in the marine geology and geophysics division, which was originally what they called. I moved from that main campus from UM in Coral Gables to the Key Biscayne campus for the Rosensia School of Marine and Atmospheric Sciences campus. And that's on Key Biscayne, and it's a lovely campus that I occasionally got to take my kayak out and just kind of roam around for an hour or two before going back to work.
Look how big this sponge is.
Oh,
Can you
wow.
get a nice zoom on him.
Yeah, and some of the lasers, please.
Yeah, he's 30 centimeters tall. Anyway,
Yeah.
continue.
Oh, it's really fascinating here. And 5 years later, I am considering myself, an adequate geologist.
Adequate.
I got to overlay a lot of the biology that I learned and with my love of marine sciences into what I do on a daily basis. And in just a few weeks, I go back and I defend my thesis, and we'll see if I get to actually finish or not.
You'll finish. Get out of here with your nonsense.
But I'm very fortunate to be able to be here also with the Okeanos and explore this underwater realm and be part of the whole mission with you guys at home and get to explore all of this at the same time you do. So this telecommunication is really extraordinary that we can talk to you in real time and you guys can see exactly what we're seeing in real time as well as what we're seeing. And it's truly an amazing experience and one that I wouldn't trade for anything. So very, very privileged to be here.
Yeah, for real, me too. Maggie McG84 on Twitter has a question.
Ooh.
She says, a bit of a morbid question, but do any of the critters survive collection? Like the sea star from earlier, or the 2 shrimp on the sponge from the earlier dive? Is the change in pressure or the process of bringing them to the surface too much for them? So it depends on the species, right? We have these bio boxes, and they're closed. So the temperature stays pretty cold as we come up to the surface. Now we do come up pretty quickly, so the temperature doesn't change that much. It changes from the bottom to the top, but inside the bio box it doesn't change that fast.
Yeah.
So we don't really get too much water mixing inside those bio boxes. The problem is if we were to collect fish, right? So some deeper fish, not all, have a swim. Well, some fish have a swim bladder. And what that is an organ inside their body that helps them maintain buoyancy underwater. So the shallower water fish and some deep water fish have air in that swim bladder. Some of the deep fish have oil in that swim bladder. So the ones that have oil in that swim bladder, it's not a problem to bring them up to the surface. They do not have the swim bladder expand. So if you were to take it has to do with pressure. So if you're a scuba diver, you'll understand this, right? So if you take a balloon and you take it down to depth and you blow air into it, you tie it off and you let it go. As it comes up to the surface, it will expand because there's a reduction in pressure as you come up through the water column. So it could expand so big that it explodes, right, underwater. So it's the same idea with a swim bladder. So fish are able to regulate the amount of air in that swim bladder, but not quick enough if you were to take them from depth to surface. So a lot of the times we'll if we're working with some fishery guys and they're doing like grouper trapping, they'll actually dive down to 90 feet and poke the swim bladder. And this is brutal, but they'll poke the swim bladder so that the air can be released. So then we bring them back to the surface, they are not choking on their own swim bladder because it's gotten so big. So it actually prevents them from passing away. Yeah. And then we return them, they tag them and put them back at the bottom of the sea, right? So they can actually survive. So they've done studies with that, and they just kind of hang out in the bottom for a little while until they feel better, and then they take off. Right.
But the other organisms that we collect, like the sea stars or the sponges stuff, usually are alive.
Yes.
That you do that. Not for long, sadly. Yeah. We end up preserving them.
Kind of our fault, though.
Yeah. All in the name of science.
Yeah.
We don't do this kindly or for fun.
Yeah.
But...
Yeah, so it
depends on the organism.
Yeah, so sea stars and invertebrates don't have that swim bladder problem. So coming from depth to the surface is not really an issue as far as pressure change, but temperature and salinity changes can cause a lot of stress.
Also,
being collected is very stressful.
Yeah.
So we have had some midwater animals sucked up through through the tube, and they're able to bring them up and see them in the fish tank and take video of them swimming around.
Yep.
But eventually they make it into ethanol, into a jar, and are labeled, and they get put in the Smithsonian. So whether they come up dead or not, eventually they're going to end up sampled.
Yep. And sometimes those organisms release, due to the stress of being collected and transported, release some sort of mucus thing that does end up also potentially disrupting the actual sample in itself. So it's one of those things that we do have to always keep in mind, and we try to minimize any sort of stress on the organism on
Yes.
itself.
And we preserve them quickly.
Yes.
So it's very very fast, and just immediate, as quickly as possible. Keep in mind that anything evolutionarily less than a reptile, less than a bird, doesn't have pain receptors in its brain. So if that makes you feel a little better.
Yes, it does.
They can feel pressure, but they can't feel actual pain. So that helps a little bit.
He's another one of those tripod fish.
Mm-hmm.
We've seen 2 previously on this dive. We haven't seen any on any of the other dives that we have there, so it could be a direct association to a lot more sediment-dominated environment rather than dense coral frameworks or coral rubble. Maybe it's not as comfortable to sit down on, or maybe it simply doesn't prefer it because it's also the type of prey that it eats is not available there. We're not entirely sure. I've never seen one actively hunting or eating anything. Have you?
No, I have no idea what they eat.
Ooh,
They're really
it's opening its
still.
mouth and closing, probably pushing some water through its gills there so it can stay stationary, because at the bottom of the seafloor there's a large scarcity of food that comes by, so a lot of the organisms here need to preserve their energy and not expend it, which is probably why the pectoral fins have been modified with its tail fin to stand up, which is why it's called a tripod fish. It touches the seafloor at 3 different points, and using and saving a lot of that energy in order for when food does come by, it's active enough to actually grab it and potentially eat it as well. So, and there is a deep sea class going out now. They're out at sea, and they're actually watching us live while they're multi-core the San Diego trough. So that's really interesting at 1,200 meters water depth.
1,200 meters. I thought you was telling me 1,200 mountain time. Like, what's happening? No. I'm so confused. That's what happens when you live in California and you're constantly going back and forth between times. It's like 1,200 M mountain.
Yeah, so hey, big shout out to those guys. Awesome, where you get to core some of these samples. I wonder what that'll bring up. I wonder how long the core is going to be if you're doing a whole drill core ship or using gravity cores. So big shout out to you guys while doing serious multitasking. We have more of these squid around. They've been more of a nuisance than anything else, stirring up some of the mud in this area. We have about 40 minutes left of this dive today.
Which means enough time for 3 more chocolates.
Maybe. Ooh, well, all the squid just darted off randomly. Is something behind us?
I know.
Do we see anything in
totally
serious?
What's behind me?
This is a great view. If you look at feed 2 from Sirius, you can see D2 along the seafloor there, and you can start to see a lot of those kind of small pock marks that you kind of get to make out a little bit in the lighting that's coming off from D2. And all those pock marks are little volcanoes and depressions of sedimentary features that is actually being bioturbated by the organisms that are burrowing into the sediments, and that they're creating a really intricate tunnel network system. And it's either crustaceans or some fish that are doing this, but it can be a multitude of different organisms that go ahead and do this. And while they're burrowing their tunnels, they push out this sediment, and that sediment ends up piling on top, which makes it look like little volcano features. And this can actually be preserved in the rock record. So we've seen this quite a few times, at least in some of the geological field trips and whatnot that I have gone to. This is actually quite interesting to be preserved, and you can actually, due to the consistency that these tunnels are usually built out of, usually find mud along with some of the mucus that's produced from these organisms. They go ahead and tap these walls to kind of form like almost like a cemented wall of these tunnels to make sure that they don't cave in. And you can also have this preserved in the rock record as well. So we get to see how sometimes these can be 2-3 meters deep and several meters wide. So it's a really intricate system. Another way to do that in situ for shallower environments, or for, as Stephanie said earlier, even for, they do this also for ants, is that you can pour resin or silver or something into one of these network systems, and you can actually then dig it out once it's all dry and set, and you can actually see the overall complex network system that a lot of these different organisms on land or in the marine system actually work.
Yeah, I remember one of my teachers doing it with ghost shrimp when I was in college, but it was shallow water. And she had like this, like, resin you'd mix together and it would solidify and work underwater.
Yep.
And then she'd dig it up and have this big, intricate.
Yeah,
I think she did her PhD on that.
Okay. We do a lot of that as well if we want to preserve some of the sedimentary structures that we see, like in the intertidal zones with a lot of those ripply structures or whatnot. Like, how do you preserve that? Well, there are certain resins that you can actually block. If you put like a wood plank down and you make a little block on its own, you can then infill it with this kind of resin that solidifies when it touches marine water and preserves everything that's exactly as it was as soon as the resin was poured onto it. So we actually have sedimentary samples of the tidal zone, intertidal zone, and right up at the shelf break, or at the tidal break rather, for a lot of these mechanisms. So we can see and learn in the classroom what it is before we go outside and try to identify them for ourselves.
Yep, that's continued bioturbation on the bottom. So, Lisa, what kind of cores are you doing out there?
You can say geology.
Geological cores. What kind of geological cores are you doing out there? You can chime in and tell us on the chat room.
Well, off San Diego, there's some carbonate, a lot of tectonic influence there, so you're seeing a lot of siliciclastic stuff, and it can go really deep, really fast. Like they were saying, like right off the coast of San Diego, they're already getting a core at 1,200 meters, and that's far below the depth that we are at today. We're about
twice this.
Yeah, we started at about 640 meters, so think almost twice of what at the depth that we've been diving at today. So potentially they could be looking at some geologic aspects, if it's a gravity core really quick that they're pulling up, or maybe some, hopefully trying to find any associated biology with them. Another quick shout out to the South Carolina Aquarium before they tune off from watching us today. We really appreciate you guys joining in and watching and discovering, helping us discover along the seafloor of what we're doing here today. So we want to thank you all for tuning in, and we'll be here for the next few days as well, exploring this particular area, potentially the Portales Terrace as well, and maybe the Agassiz Valley further down and past Key West before we officially end this cruise in 4 days?
The port. We come into port on the 20th.
Yeah, on the 20th. So we have potentially 3 more dives to do. So please come back and tune in. We have still tomorrow, Monday, and Tuesday when we will be diving.
Yeah, tomorrow I think we're going to try and do the inside and the outside of this Portales section of the HAPC, the HAPC that we were diving in almost all week. We're going to try and do both inside and outside in one dive, so cross over the governmental boundary, but likely we will see nothing change because animals don't care about that. Hard bottom doesn't care about it. So we're planning on that tomorrow.
Do you want to explain what the HAPC is?
So habitat area, the HAPC or the HAPC, or the habitat area of particular concern, is a type of marine protected area. So the one that we're going to be in tomorrow is part of one of the largest deep water coral habitat areas of particular concern in the United States and I believe in the world. It was closed in 2010 based on the research that my boss John Reed submitted using a combination of 35 years of submersible diving and multi-beam data to predict the likelihood of hard bottom and therefore possible Lophelia coral reefs in the area. So in the HAPC, you are not allowed to bottom trawl. However, you are allowed to surface trawl. So it's specifically made to protect the benthic, slow-growing, precious corals. These deep sea corals. I think dive 6 was our Lophelia dive.
Yes,
I think it was
so.
dive 6. If you go back to dive 6 in the data, you'll see it. The Lophelia mounds we were on,
And those were outside.
but those were outside. That was towards the eastern side
of the Blake Plateau.
Yeah, and they were outside the HAPC.
On newly acquired bathymetric data.
Correct. That was acquired by the Okeanos 2 weeks before. So that's basically the HAPC is there to protect the bottom. There are sections that are called allowable crabbing areas, and that's where there's little sections inside the HAPC that look more like this, that look more like sand bottom, where crabbers are allowed to fish for golden crab. So there are sections where they are allowed to do that, but it's limited. So tomorrow we're going to be in that HAPC.
Tripod fish.
We're going to, yes, he's cool. Look how cool. Look
His
how
tail
cool.
is, though. His tail fin is black.
Yeah, and spread open, right?
Yeah.
I like this antenna.
Yeah.
Like little alien. It's too cool. Yeah, he uses a little balancing act. He does on his fin tips.
Great. Sorry, I didn't mean to interrupt you.
That's fine.
I had to comment.
I was rambling. It's totally fine. I mean, I ramble all day, so can't help myself. That's why I'm here, I guess. I'm good at rambling. Yeah, so tomorrow we'll be in the southern part of the HAPC, going from inside to outside. So we're going to have that marked on our map so we can see when we're inside and outside.
Yep. That seems to be just about 10 centimeters long.
It looks like there's something in there. Yeah, definitely.
Ooh. And you can just see how fine the sediment is. As soon as the ROV touches on the bottom, it just kind of wafts up this huge cloud of finer grained sediments. Where in different areas that we looked at, especially in the Blake Plateau and further up north, we didn't have that. We had a lot of coarser material. We had every now and then there were some muddy sediments, but for the most part, you could really see every individual grain when there was a zoom in on there. So it was a lot of coarse grained material, and that is composed of a lot of skeletal material, and that's all calcium carbonate oriented, meaning that it's composed of the same skeletal material as your own bone structure. There just happen to be different mineralogical properties in them, and there's 3 main mineralogical properties. We have aragonite, calcite, and dolomite. And aragonite is mostly composed, is a lot of what your skeleton is composed of, as well as coral skeletons and different smaller organisms. And then calcite is another one of those major ones where it's usually precipitated out of the seawater column. And it is also associated with a lot of the skeletal material from different organisms that are there. And dolomite is rarely ever formed right out of the seawater. And carbonate has to be formed, at least in the marine environment, has to be formed in the seawater in supersaturated zones. That dolomite is usually the diagenetic effect from other aragonite or calcite minerals to form dolomite. So it's a reworked system that has then some sort of effect in it, whether it be burial heat difference in water type, where if you start to have meteoric water, that means rainwater or fresh water flowing through a carbonate feature, it can start to impact the overall mineralogy of it, forming that dolomite, reworking the actual carbonate systems and forming that dolomite system. And a lot of these skeletal fragments are composed of pteropods, foraminiferous shells, and those are little tiny, we refer to them as bugs of the ocean, where they just float around in the water column. And we actually can use them. There's different species that are known to float at different levels within the water column, and they collect the isotope data that's in that water column into their skeletal structures. And if we know around what level of the water column, of what depth the water column they usually live in before they die, we can then analyze the isotopes and refer at what point in the water column they were living at, how they were living at, and what that structure of the water column actually looked like. So they're very important to the overall systems as far as contributing to the sediment factors here, and they can also be microscopic size, mud-sized particles, as well as coarse grain that's larger than 250 microns. That's about 2 millimeters in length, and that's quite, in geologic terms, that's defined as a coarse grain sand. And they compose a lot of the material in this region, as well as all of that muddy material. So the muddy material can also be breakdowns of these skeletal fragments. So you might not have whole bits of them, but definitely smaller chunks that just get reworked, maybe in the current systems, and what have you. And here you can see more of these depression features that are likely events of bioturbated micro or macroorganisms in the area. They form little tunnels, intricate tunnels down underneath the subsurface here.
Yes. This whole dive pretty much looks similar to this, with lots of these little mounds and pock marks, both active and filled in. Within these small holes, we've been seeing a type of crab that looks similar to the golden crab, but much smaller, and a little bit hairier. So likely the same family of crab, but a different genus. We've also seen pretty commonly these carnivorous sponges. We've collected 3 today. They are a very rare sight in general. However, they're, I wouldn't say common, but present here. They are the most common species of sponge, I think. Maybe Hylanema is the only other one we've really seen today. That's the cotton candy ball on a stick. Is really the only other species of sponge we've seen. Otherwise, it's fairly barren. It looks like we've lost all of our swimming ilex squid. We have a couple of these tripod fish. We're crossing over one right now. We've seen a few of those today. Yep. And these pock marks are filled. A lot of them are abandoned. This one looks like it's active. This one right here at 9 o'clock, a larger one. Yeah, so that looks active to me. There's also another one in the distance. That's closer to noon now. You can see the walls of it are still maintained. Yep. And even the one behind it, they might be attached. Yeah, so this is an active burrow. So somebody's probably actively living in there. I see something. I see the tail of something.
I think it's maybe a fish taking advantage.
Yeah, it looks like a Lamanema tail. If I had to venture a guess. You can see how thick the sediment is, though. Right here. I mean, it's deeper than that, right? Yes, this could be a hole from a tile fish, sand tile fish, or a a golden tile. They live out in the sediment. Could also be from Acanthocaris, the blind lobster. I saw one at the very beginning of the dive, but I haven't seen one since. And usually these little holes sometimes have a back door, depending on the species.
Yeah, I mean, we've seen quite a few different species. Overall. It's not a biodiversity hotspot by any means, but we have seen a variety of species in this area. Still wondering what can really form these large depression features that lead to the tunnels here. Could be that tile fish, could be something else.
Yeah, I really don't know. I don't know if we'd ever figure it out. Maybe if we put a still camera down here and just watched for hours on end and see what came out of it.
Yeah.
Out of an active burrow. ROV's pretty loud and very bright, so I would imagine we're probably scaring whatever it is off.
Yep. So, Steph, I have a question for you now. What has been your favorite thing that we have seen across these dives? And that does not count the Lophelia reefs.
Probably the picniconid, the sea spider with babies.
Oh, that has to be a good one.
Yeah. That's a good one. How about you?
So
I swear if you say sand waves, I'm going to lock you out of the room.
Okay, as important as those features are, I have to say the biologist in me is still totally in love with the fact that the pycnogoid with the babies was surrounding it was definitely a major highlight from this. I loved seeing the isopod, and I've loved seeing and learning all of the different associated biologies, because even though I have studied biology throughout my undergrad career, in graduate school, learning about the cold water corals and their mounds that they developed through time, I'm considering more the geologic aspect than the biological aspect. And I'm sitting here working next to you and learning about all the different kinds of corals that there are, different mechanisms, different DNAs, different clades, different associated biology has definitely been a highlight that I hope you can experience through a textbook. And being able to be here and experience these organisms firsthand and working with them in the microscope and learning more about how the DNA structure works and all of that definitely has to be part of my favorite portion of this entire trip. Being on the ship and working hands-on with the environment and being part of something that's potentially a whole new species and something we've never seen before is definitely one of the highlights for me on this cruise.
Yeah, for sure. I really like the idea that I can look at something and say, I don't know what this is. And then within 2 minutes, someone's ringing the phone telling me.
Yeah, and
so we don't usually get that. So I get to go to sea 2 or 3 times a year regularly, right? So I've got, I usually have a couple fish people on the boat. We've usually got John on the boat. But, there's some things we run into and we're like, I don't know what this is, right? A lot of the sponges, because sponges are impossible. So a lot of the sponges, I don't know, it's a brown sponge. So we just give it some name. We had one that looked like bubble wrap. So its name is the bubble wrap sponge. I don't know what to call it because I don't know what it is. But if we were out here on the Okeanos and I saw the bubble wrap sponge, I could say, oh my gosh, I don't know what this is. And then Chris or Shirley would be able to chime in from land and tell me. So that's a huge, that's a huge benefit to being on this cruise.
And those look like fecal pellets from a sea cucumber.
Right? And a Hylanema stalk.
Yeah,
it's abandoned.
Some of the silicate stock that's there. So silicate is a very different material than the surrounding calcium carbonate. Silicate does not dissolve as easily. It has an entirely different mineralogical structure, and it's a completely different element than those composed of calcium carbonate. It's siliceous. If the silicate, that's why it's called more silicate materials. Of these spicules, they do not dissolve, at least very easily, and they stay around quite often. This is one of the major things we do see in sediments when we collect them. And this is one of the major components that, besides being almost all calcium carbonate oriented, whether from broken up skeletal fragments or precipitated sediments, that we do see a lot of these sponge spicules, and that contributes towards the silicate that we're seeing in the environments, and how much we're seeing, the density that we're seeing, possibly different kinds of spicules that we're seeing all relate, and we use also that information for the geologic rock record of where we are potentially are in an entire system, where we're more likely to see more of these silicate precipitating sponges. It all plays in a major factor of what we're looking at and what we're learning at. So despite my profession being from geology, being in calcium carbonate, we do have to learn a lot of these other factors that contribute to the actual sediments and what we're looking at here, because it's not strictly what you would expect it to be.
Yeah, and I wonder, I mean, because the Hylanema sponges are somehow precipitating silicate, right? And making those long stalks. So I don't know how that works.
Yeah.
I would imagine it's kind of the same process as corals make calcium carbonate skeletons, but I honestly don't know. So that would be something cool to look into. I wonder if Chris knows how that works.
Hola, my friend.
Hi.
How are you?
Good.
I'm here, inspired by your amount of energy that never ends.
Yeah, who knew I could talk for 8 straight hours?
I don't know exactly, like how long it takes to excrete those very long features.
Mm-hmm.
I promise I'm gonna look into it.
You're looking how that works.
And how that works. Spicules are siliceous spicules are secreted in the case of Exactinelli and Demospongia along proteinaceous filaments. So there is first a proteinaceous filament inside of a cell in which silica starts to be secreted along, and then the species mold, it gets extruded from the cell, and there are other cells that deposit more and more silica along the proteinaceous filament. However, I don't imagine how this occurs in these very long species. If there is a growth, it's always at the base of the sponge, because I imagine that the first part of the root is the oldest part. So it's a very interesting question, and I promise I will look into it, because they probably it has to be studied. Maybe not the process, because think about to keep alive this type of organism, deep water organisms. I don't think that this has ever been done. But I would look to see if there is anything known about it, okay? Anyway, it's really incredible. And for me, those pink isopods on the base of the stalk were a half point on the dive today, the same that the starfish eating the top of the carnivore sponge.
Mm-hmm. Awesome.
Absolutely. And here we have another one of those armored sea robins.
Yep, with his modified barbels. Looks like he's gonna sneeze.
Wow.
Yeah.
Oh, wow.
Yeah. He's so weird, isn't he? He's so weird. Look. Let's see what he does. Oh, that was lame. It was very anticlimactic. Maybe he was yawning. I don't know. Are you going to be around tomorrow, Chris? Are you busy? Maybe she ran out.
I'm here. Oh, sorry, I'm here.
Oh, okay.
I will be with you guys tomorrow.
Oh, okay, great. We should be. And Shirley's coming too?
She will be Monday, Tuesday with us. So.
Okay. Yeah, we should be on hard bottom tomorrow, so we might, likely see more sponges. So
Well, this was
Keep you
pretty
handy.
exciting and pretty happy, I would say.
Yeah. Awesome. Thank you, lady.
Thank you. And I really appreciate all of the input that you've given us towards all the different sponges and the different collection items. It's been invaluable, and this is one of the main reasons why this kind of work is so important and our telepresence is so important, because onshore scientists like you can chime in and really give us the information that we need that we might not have ourselves on hand here. So big thank you towards that.
Well, it's my pleasure. Thank you.
We love you, Chris. Chris.
Oh, it looks like we have a blind lobster here.
Oh, nice.
And another carnivorous sponge. Wow, we're in abundance here.
Right. Of course, 10 minutes before we get off the bottom. Typical. So this, I do not think, is a blind lobster. This is a different species of lobster. He's pink, but he's not a Acanthocaris. His front claws are way too big. So it's not Acanthocaris, but I do not know what it is. It may be an Alpheid, a type of snapping shrimp. I don't know how big he is. When we can I get lasers on him? I don't know if... So he might be one of the creators of these burrows. He's definitely covered in sand and all these long hairs.
Yeah, it seems like the sediment is really trapped kind of on him rather than just resting on him. Or it, rather.
Yeah. I'd like to see his claws in the front, but... Oh, he's pretty big, 10 centimeters wide.
Yeah. There's another one of those carnivorous sponges out there.
Cool.
Ooh, nice.
Yeah, he's really hairy. See all the accumulation of like, he's like a bear, right? It's like covered in fuzz.
Really fuzzy. And it's not all sediment, that's for sure. It just looks very fuzzy all around. I haven't seen anything like that before.
So I've seen the crab. There's a crab that has really fuzzy claws.
Yeah.
But I think they live on the sea vent. I mean, the vents.
The hydrothermal vents?
Yeah, I think so.
I don't know. Yeah. I don't know. Well, cool. Thanks, pilots, for that other view. We have another question on Twitter from the Good Doctor. Is there any data about accumulation of human-made pollutants, heavy metals, or other toxins within the organisms that have been collected? Is there any theory about how these chemicals may impact deep-sea inhabitants? So I'm not familiar with anything that's been done as far as the individual organisms that we've been collecting, and that was on Twitter, Stephanie's Twitter. And I'm not sure about anything that any workup has been done by the actual animals themselves. There is a lot new studies going on for microplastics that's been seen within the organisms, digestive systems, or accumulations on their outer shells, or anything of those kinds. But physical chemical toxins, I'm not aware of that has been doing that. We have talked in previous dives about how climate change is definitely impacting even the deep sea ocean. The CO2 gas exchange that's been incorporated into the water column in the ocean. The current systems are driving a lot of that then CO2 rich water down into the deeper ocean, and as ocean acidification occurs, there's actually different layers in the water column that are more saturated in terms of calcium carbonate than others, and that with ocean acidification, that layer is actually being driven shallower. So less and less of these carbonate precipitating organisms can live in deeper parts of the ocean. And we're talking in the general thousands of meters range rather than in the hundreds where this area is here that we're diving at today. So if you tuned in the Pacific region, that was last year's or the year before, expedition dives, the ROV went down to 4,000 meters, and this is well below the area of where the saturation levels are actually occurring. And while we do see some carbonate precipitating organisms, they're very sparse, and we're not seeing them in abundance like we do in certain areas, like that cold water coral mound that we came across of Lophelia reefs and things like that. We're not seeing it as robust anymore. And with ocean acidification,
Ooh, look in that hole.
Looks like another blind lobster.
That's a Acanthocaris. Okay, thanks.
No, never mind. We are actually experiencing where these sea water level saturations are actually getting shallower, and that's a big problem for a lot of the organisms that have been living possibly millions of years at a certain depth that they have been living at. And so it can affect them in that general respect, where they're actually like crustaceans, where a lot of their carbonate shells are in the outer portion. There's no tissue to cover them and protect them from the dissoluting environments around them, the more acidic waters. They're starting to dissolve, and they can't precipitate these shells, and therefore they're dying. So
there's also been some studies done on marine mammals because fat can be an absorbent of toxins. I do not know much about that, but there has been research done on that. So definitely something to look into. If I can find anything, I'll post it to your Twitter feed. But yeah, microplastics is a big problem. I know that in the Pacific, I believe planktonic plastic outweighs natural plankton 6-1. So please recycle. Yeah, and there's the Pacific garbage patch, which is, it's pretty bad. So we need to figure out a way to clean that up, I think.
Oh, are we officially off bottom
Yes.
now? Okay. Well, thank you all for tuning in today. We greatly appreciate you joining us today, and we hope to see you again over these next few days while we finalize this cruise here with us, and you get to explore then with Steph and I around here. And for those that want to stick around for the post-dive call, we will be tuning into that dive call at approximately 4:20. So that is 16:40 Eastern Time.
16:20.
16:20, I'm sorry. I don't know why I keep doing that. 16:20 Eastern Standard Time. So that is 20 minutes after the top of the hour. And we'll be back for that post-dive call. But thank you all for tuning in with us today. We hope to hear back from you tomorrow. We welcome all questions and emails and suggestions that you guys have. Please use the social media aspects. We have 2 different Facebooks. Noah has 2 different Facebooks that you can contact and post questions to, at Ocean Exploration